we find cardiac arrest situation, when we found a victim of cardiac arrest, as soon as possible we have to give the victims a basic life support, which is CPR. And then when indicated, we should immediately get the victim a defibrillation. When the victim's condition is going to stable, immediately transfer him to the hospital to get further treatment. The treatment can be catheterization or treatment in ICU. Next. This is the warning system that we have formulated when there is cardiac arrest condition.
Next, okay. Next, okay. This, Gigi.
And we have this design of some system work to apply this prototype. The first, we have model heart rate sensor and power supply unit that transfer the energy to microcontroller ESP32, and then the microcontroller transfer the signal with Bluetooth Low Energy to the smartphone. Next. The diagnosis test is divided by two, is the tool design state and prototype trial phase. At the stage 1, the test was carried out of healthy participants placed in Bapolkes Batam. In stage 2, the test was conducted on participants with dysrhythmias, location of Abdul Malik Hospital in Province of Lampung. Next.
Next, okay.
And
that is okay. This
the prototype tool. The results of consultation with cardiologists, electronic expert, and reviews. The design focus on detecting pulse accurately, and the result of the pulse frequency are more accurate.
That is okay. That is the certain discussion.
And here we come with the result and discussion of the research. In addition to the result of statistical analysis calculation, it is necessary to consider several conditions that affect the effectiveness of the prototype in sensor of pulse. The one.
That is conclusion.
And we have come to the conclusion. The first, the pulse sensor prototype that has been designed has a sensitivity value of 98.5% with a specificity value of 50%. The pulse sensor prototype design is connected to a smartphone in real time. And last, the bulk prototype will automatically activate an alarm and provide guidance on performing basic life support if it does not detect a pulse. And here our suggestion for the next researcher, the performance of the sensor based on photoplethysmography, which detects change in blood flow volume. It is necessary to carry out further tests that consider skin color, especially dark colors, skin covered in ink such as tattoos. Sensor performance is based on a change in blood flow volume, so it is necessary to consider factors that affect blood circulation such as blood pressure, total body fluid volume, and elasticity of blood vessels. Further research on advanced life support states by establishing a connection between the victim's smartphone and PKN 119 Ministry of Health Jakarta are a service center where the victim registered for healthcare. For research also can eliminate the inhibiting factors for censor work so that censor results are more accurate for everyone.
Okay, finish, Mr. Kiki. That is a picture test my researcher. Okay, thank you. I will, I think finish my presentation, Mrs. Siti. Please welcome.
Thank you very much, Mr. Purbianto, for the presentation. Ladies and gentlemen, the second presentation will be delivered by Mr. Ahmadiani. We invite Mr. Ahmadiani to deliver the presentation, please.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh.
Yeah, thank you very much for the master of ceremony, the Honorable, the Director of the Polytechnic of Health of Banten, and also the Honorable of the Director of the Polytechnic of Health of Banten, and also the Honorable participants in this event. Please allow me to introduce myself first. My name is Ahmadiani. I'm part of the Polytechnic of Health of Banten, from the hypertension prevention and control research center. Please allow me to present the non-communicable disease that we research together in this institution. Yes, our recent research is about the infusions of Hibiscus subdariva and Cucumis sativus show potent inhibitory activity towards angiotensin converting enzyme. The basic background from this research is that the patients with hypertension require drug therapy, especially with to control their blood pressure and maintain normal conditions. Several types of drugs used to treatment the hypertension usually belongs to angiotensin converting enzyme inhibitors, which work by inhibiting the conversion of angiotensin 1 to the angiotensin 2, which is a vasopressor. However, the antihypertensive drugs have limitations because they must be adjusted to the patient conditions, especially those with contraindications. So in addition to the use of the synthetic drugs, in the long term, they can cause undesired side effects, such as symptoms of hypersensitivity reactions, such as itching and also the upper respiratory tract infections. So because of the side effects and also those limitations, we developed these alternatives for the patients, especially to conduct the non-pharmacological therapy. We go from the other medical practices in Indonesia, which people usually use Hibiscus subdariva, or we know about this flower called Rosella, and also the Cucumis sativus. We find it every day. We call it mentimun or timun. So these two plants have phytochemicals composition that can attract our research to develop the phytochemicals modifications. So with these various functions or activity of these two, one of them is the S inhibitor activity. So we go from this basic theory. So the objective of the research is directly to measure the S inhibitory activity of the infusions. If we find out that the infusions have the activity to inhibit the hypertension, especially in this case is the enzyme, so we can conclude that the infusions of the Rosella and also the Timun can be functional drinks. So the material and method that we conduct before the SI kits, chemicals, and instrumentations, we get from the laboratories, especially for the enzyme kit, and also the captopril for the control positive. The positive control we use here is the easily find medications for patient with hypertension. And also the chemical reagents used were the analytical grade. And the instrument we use is the microplate reader from Thermoscientific to measure the inhibitory activity. The collection and identification of plant materials we conducted at Balai Materia Medica in Batu, Indonesia. So the dried calyces of Sabdarifa and also the fruit of Cucumis sativus we collected in March to July 2022, and then it were identified and then authenticated taxonomically in Balai Materia Medica. And then the preparation of the plants materials, we use the common method where we rinse to remove the contaminants first, and then dry the plant materials, and then the dried materials we ground into coarse powder and then stored in the clean container until we prepare the infusions. This is the preparation of the infusions. We separate into three parts. The first one, we powdered the powder of Sabdarifa, weight into 10, 20, and 30 grams each. And we put into a conical beaker and we coated it with HS1, HS2, and HS3. And the first cut sativus also was weighed in similar procedure, and the sample were coated with CS1, CS2, and CS3. So the combinations of the Sabdarifa and sativus also prepared with the ratio of 1:1, 2:1, and 1:2 ratio with the total of 30 grams each. And also we prepare those combination with the coded HC1, HC2, and this HC3. And then every sample was steeped with 30 milliliter of hot distilled water and shaken gently until all plant materials were soaked for 1 hour. And then we filter it with Wattman number 1. So the filtrates stored in the clean plastic containers until it being analyzed. And then we also conducted the phytochemical screening for the phytochemical compounds of flavonoids, alkaloids, saponins, tannin, and steroids, and also terpenoids. The final work will be the determination of DS inhibitory activity. We use the colorimetric method using the DS kit WST36 following the manufacturer's manual, and use the captopril 25 ppm as the positive control. We prepared also the extract or the infusion of those three combinations. The absorbance was read at 450 nanometer using thermo multiscan, and the inhibitory activity in percent were calculated. This is the result of the phytochemical screening. From the table, we can see that there is no steroid found on these three kind of infusions. Also, flavonoids we cannot found from those three infusions, but we can find saponins on the three infusions. From this, we can conclude that during 1 hour preparations of the infusions, maybe the process to extract all of these phytochemicals is not enough. So basically, the easily extracted compound from the infusions was saponin. Yeah, because we prepare the infusions in aqueous base, so flavonoids and steroids were not detectable in all infusions, but saponin were found in all infusions, which indicates that saponin were successfully extracted from those 3 infusions. And alkaloids were absent. We can see this, that only Cucumis sativus contains the alkaloid. So in the combinations, we found the positive value here. It came from the Cucumis sativus. And also for the tannins, we can see that the HS infusions contain the tannins, but no in the Cucumis sativus. So in the combinations of infusions, here the positive was came from the tannins content from the Hibiscus subdariva. The terpenoids, as we know, contained in the Cucumis sativus. So this can confirm the findings, and also the HS infusions contain the. So the presence of secondary metabolites in every infusions depends on the effectiveness of the extraction process, which is in this case is the infusion process using the steep water, the hot water, 90 degrees Celsius in 1 hour. So we believe that the extraction time and also the polarity of the solvent influence this result. So all samples were soaked in hot distilled water for 1 hour, and this process was probably not long enough to extract metabolites from the plant materials. So in addition to this result, the solvent we used, which was polar, had limited ability to extract non-polar or semi-polar metabolites due to the difference of in polarity. However, the higher amount of plant materials may affect the metabolite yield extracted from the infusion process. And this is the S inhibitory activity result. The blue one was the HS infusion from the Rosella, and the orange one was the CS infusion from the cucumber. So we can see from the same concentration that the CS infusions has the higher activity than the HS infusions. Although the captopril as the positive control remains the strongest between all of them. The strongest activity from the Rosella was obtained from the 30 gram per 300 milliliters, which was 88% inhibit the enzyme. And the CS infusion was 92% inhibit the S enzyme in the same concentration. However, for the captopril, it has the highest activity with 97% against DS activity. And this was the result of the combinations of the Rosella and the Mentimun compared to the highest concentration of Rosella and also for the Mentimun. So the combination shows that the inhibition activity increased. In this case, on the HC3 formula, the combination of 1 Rosella to 2 cucumber has the activity of 96% towards the angiotensin converting enzyme. Nevertheless, it has the strong activity among all of infusions. Although the strongest activity showed by the HC3 is still lower than the captopril, which is 97%. So we can conclude from this finding that infusion of Sabdarifa, Sativus, and also the combination of them show potent inhibitory activity towards angiotensin converting enzyme. And also the strongest inhibition activity is demonstrated by the infusion of HC3, which was close to the captopril. Since the dose of Sabdarifa used in this study was far below its median level dose, and the cucumber also has no dose-dependent side effect, the infusion HC3 is potential as a functional drink for managing hypertension. The challenge from this study may be how we in the future develop the way or method to lowering acidic state of the infusions, because as we know, Roselle infusion contain acidic aqueous solutions, so it still maybe can affect some people with gastroesophageal problems. So we don't recommend this for the people who has gastroesophageal problems to consume the infusions. And also in the near future, we are planning to do some research about the side effect of these infusions. So maybe that's all from me. I hope this finding may give some information to you guys. Thank you for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh. I'm giving it back to the MC.
Thank you very much, Mr. Ahmad Yani, for the presentation. Ladies and gentlemen, now we are moving the main event of the webinar. The session will cover 3 topics delivered by 3 remarkable speakers. The first one will be Professor Yuven Wong, who will deliver the topic of miRNAs as potential biomarkers for hypertension. This fourth session will be moderated by Ms. Jiska Mariska. To moderator and speaker, please take your time. Ladies and gentlemen, we apologize for the delay. Ladies and gentlemen, now we are moving the main event of the webinar. This session will cover 3 topics delivered by 3 remarkable speakers. The first one will be Professor Yu Feng Hung, who will deliver the topics of mRNAs as potential biomarkers of hypertension. This first session will be moderated by Sister Mariska. To moderator and speaker, please take your time.
Okay, thank you for the nice introduction for the Master of Ceremony today. Assalamualaikum warahmatullahi wabarakatuh. Greetings and a very good day for everyone. Suara saya jelas terdengar? Greetings and a very good day for everyone, especially to our honorable speaker, Professor Yufan Pung from Malaysia. I hope you are already joining us today, this morning. I would like to thank you all the participants for finding time in joining us today in the first seminar session of Current Research in Non-Communicable Diseases webinar held by Polytechnic of Health of Banten. As we all probably know, non-communicable diseases are not passed from person to person. However, those diseases such as heart disease, stroke, cancer, diabetes, and chronic respiratory disease are collectively responsible for the majority of deaths worldwide. So this situation calls for urgent action and plan to better manage NCDs and reduce the burden of NCDs subsequently. Now, as the world of medicine has now entered this new era called precision medicine, where we take into account a person's genetic makeup, personal environmental exposures, and lifestyle, there is a growing interest in the potential of genetics and genomics as both the diagnostic and therapeutic tool. And today we are privileged to have Professor Pung, all the way from Malaysia, to share and give us insight on how microRNAs serve its role as biomarkers in heart disease, in both diagnosing and managing the condition. Mohon untuk panitia untuk menampilkan CV dari Profesor Pung. Akan saya bacakan. Boleh minta tolong panitia? Oke, sambil menanti panitia untuk menampilkan CV Profesor Pung, pembicara kita pada pagi hari ini, izinkan saya untuk membacakannya saja sendiri. Professor Pung, she is from University of Nottingham, Malaysia, has been a stellar academia and researcher with an outstanding educational background and training, on also a handful experience in teaching, conducting research, and also publication. I could go on and read all the achievements that she made, but I'm afraid that we don't have any much time because the time is so limited today. Apakah Prof. Pung sudah bersama kita, panitia? Hello, Prof. Are you here with me already? Mohon informasinya dari pihak panitia, apakah sudah ada Prof. Pung bergabung?
Hello, Ms. Diske. Yeah, I got the news from Professor Pung that she has delay to connect with us this morning.
So maybe
she asked for additional time to reconnect her gadgets to the link.
Sure.
Thank
Thank
you.
you.
Probably I would like to ask for the committee to display the CV of Professor Pung so that the audience can read, and I could also read it for them. Boleh tolong ditampilkan? While we wait for the Prof. Yun to join us here today, we would see the CV of Prof. Pung first so that the audience have this grasp of what kind of research that Prof. Pung did for all this time and what kind of topic that she's interested in. Okay. Prof. Pung, mungkin karena belum ada Prof. Pung, saya izin menggunakan bahasa Indonesia terlebih dahulu untuk para audiens. Prof. Yu Fan Pung adalah seorang associate professor yang bekerja di University of Nottingham, merupakan university dari UK juga, namun ini adalah cabang dari UK di Malaysia, yaitu Malaysia Campus. Beliau saat ini current research-nya memfokuskan pada mitochondrial bioenergetics to address issues related to obesitas, diabetes, hypertension yang akan beliau bawakan pada pagi hari ini, dan juga cardiovascular physiopathology. Untuk professional employments, beliau sendiri saat ini sejak tahun 2017 sudah menjadi associate professor di University of Nottingham. Sebelumnya juga sudah menjadi research assistant di National University of Malaysia and also research assistant di Genome Institute of Singapore. Beliau memiliki banyak sekali pengalaman basic education dan training di berbagai tempat di belahan dunia, mulai dari Malaysia, Singapore, lalu ada di United States, di California dan di Ohio, lalu ada juga di Cambridge, United Kingdom. Beliau saat ini juga bertugas sebagai anggota dari Malaysian Applied Biology Journal, Malaysian Proteomics Society, lalu ada American Heart Association, American Stroke Association. Jadi saya pikir beliau adalah seorang pembicara yang sangat-sangat kompeten membawakan topik mengenai biomarkers terbaru yang digadang-gadang dapat men-screening dan juga membantu dalam tata laksana penyakit-penyakit yang tidak menular, yaitu hipertensi, yang merupakan keahlian beliau. Untuk teaching experience dan coordination of courses, beliau juga merupakan salah satu pengajar dengan banyak sekali teaching experiences, research support and patents, lalu ada banyak awards juga yang sudah beliau hasilkan. Jadi memang Bapak Ibu sekalian, tidak usah diragukan lagi, insya Allah kita akan sama-sama mendengarkan paparan yang sangat menarik dan insya Allah mudah-mudahan membawa manfaat bagi kita semua. Is Professor Pung here already, Kamati? Or she's still taking her time to join us through her gadget? Mohon dinantikan terlebih dahulu Bapak Ibu ya, mudah-mudahan nanti segera setelah ini kita akan berkesempatan untuk mendengarkan paparan dari Prof. Pung sendiri.
Ms. Ziske, Professor Yu Van Fung already joined us, so we can continue the sessions. Thank
Ah,
you.
thank you. Comedy, Prof. Yu Fanpul, are you already here with us? Hello, good morning.
Yeah, good morning. I'm already in. Sorry, I didn't know you are waiting for me.
That's okay, that's okay. So I would like to restart from the beginning if you don't mind. Okay.
Yeah, sure. Can I try? Can you hear me well?
Yeah, sure. I can hear you well, and it's clearly.
Okay. So
Okay.
I'll start to share my screen. Is that okay?
Sure. But before you start your screen, may I introduce you first? Because I already introduced you before, but now that you're here, I would like to start again, if you don't mind.
Yeah,
Would it
sure.
be okay? Yeah, sure. Thank you. Okay, thank you for the nice introduction, the Master of Ceremony. Assalamualaikum warahmatullahi wabarakatuh. Greetings and a very good day for everyone, especially to our honorable speaker today, Professor Yu from Malaysia. Good morning, Prof. But it's already like almost 11 there, I guess.
Now it's 10:45.
Good. I would like to thank you all the participants for finding the time and joining us today, the first seminar session of Current Research in Non-Communicable Diseases webinar held by Polytechnic of Health of Banten. As you all probably know, non-communicable diseases are not passed from person to person. However, those diseases such as heart disease, stroke, cancer, diabetes are collectively responsible for the majority of deaths worldwide. Now, this situation calls for an urgent action and plan to better manage NCDs and reduce the burden of NCDs subsequently. Now, as the world of medicine has now entered this new era called precision medicine, where we take into account a person's genetic makeup, personal environmental exposure, and lifestyle, there is a growing interest in the potential of genetics and genomics as both the diagnostic and therapeutic tool. And today we are so privileged to have Professor Pong all the way from Malaysia to share and give us insight on how microRNAs serve its role as biomarkers in heart disease in both diagnosing and managing the condition. Prof. Pong from University of Nottingham has been a stellar academia and researcher with an outstanding educational background and training and a handful experience in teaching, conducting research, and also publication. I could go on and read all the achievements that she made, but I'm afraid that we don't have that privilege since our time is limited. So without further ado, please give a very warm welcome to Professor Pong. And Professor Pong, for the next 40 minutes, the screen and time is yours.
Okay, thank you for the chair lady for the very nice introduction. So I just want to check, can you hear me clearly?
Yeah, crystal clear.
Okay. So very good morning, ladies and gentlemen, colleagues and students. Sorry, I'm late. Okay, so first of all, I would like to thank the organizing committee, especially Prof. Ahmad Yani, for inviting me to give or to share my research experience in the current research in non-communicable disease webinar. So I'm Yu Fen. I'm from the National University of Nottingham, Malaysia. As you can see, University of Nottingham has the mother campus in UK. So it also has two other branches, one in China and one in Malaysia. So we are actually the branch of University of Nottingham located in Malaysia. Okay, so first of all, let's look at non-communicable diseases and the prevalence worldwide. So non-communicable diseases kill 41 million people each year. Okay, this is equivalent to 7 in every 10 deaths worldwide. And non-communicable disease usually comes from avoidable causes. So which means that if you change your lifestyle, environment, and diet, you can basically change the disease outcome towards a more positive prospect, okay? And non-communicable disease affect age group from 30 to 70-year-old, which is the most productive year no one can contribute to the nation and society. So according to WHO fact sheet in 2017, so they identified 5 health clusters that contribute to the most death tolls in non-communicable diseases. They are the cardiovascular diseases, diabetes, cancer, chronic respiratory diseases, and mental health condition. Okay. So can you see my slide clearly, because there is a black bar here blocking?
Oh, the black bar is the controller for Zoom, I think. You can
Yeah,
just
so can you see my screen clearly or no?
we can see the white part, but the blue part where the title is, we cannot see it because it was blocked.
Okay, how about if I move down? Can you see?
Yeah.
Okay, but it's blocking the bottom now.
Yeah, but I think there would be no writings there. I don't know. Probably.
Yeah, I think it's blocking the bottom part. I'm not sure how
Can
to
you
remove.
can you drag it all the way to the top? No.
Yeah, it's still blocking.
Yeah. Okay.
Yeah, I cannot remove it.
That's okay, as long as we get to see the white part.
Okay.
Okay.
Okay. So first of all, let's look at cardiovascular diseases. So cardiovascular diseases are the world #1 disease killer. Okay, so cardiovascular diseases are a group of disorder of the heart as well as blood vessel. So having said that, for example, stroke, ischemic heart disease, peripheral artery diseases. So these are all part under cardiovascular diseases. So as you can see here, according to the World Heart Federation in 2019, so cardiovascular disease contributed to 17.8 million deaths worldwide. This is even more than cancer. And there are a lot of risk factors that can contribute to cardiovascular diseases. So example would be like high blood pressure, high cholesterol, diabetes, obesity, and also some environmental factor like air pollution, as well as lifestyle, for example, physical inactivity, use of alcohol, tobacco, or unhealthy diet, and even kidney disease. Okay, these are some of the risk factors for cardiovascular diseases. Okay, now let's look at cardiovascular diseases in Malaysia. So in Malaysia, cardiovascular diseases are still the Malaysia #1 disease killer. So according to Malaysia Mobility Survey in 2019, so cardiovascular, the ischemic heart disease itself contributed to around 16,000 deaths worldwide. In Malaysia, sorry. So approximately 1.7 million Malaysians suffered from 3 major risk factors, as shown here. And then approximately 3.4 million Malaysia suffered from 2 major risk factors. So either they suffered from diabetes and hypercholesterolemia, or hypercholesterolemia and hypertension, or hypertension and diabetes. Okay. So in the past, right, during my postdoctoral training, either in United States or in UK, so I work on cardiovascular diseases as well as the risk factor. I'm trying to understand what are the causes leading to cardiovascular diseases, as well as trying to find preventive measures and treatment, okay, to basically regress the progression of cardiovascular diseases or the risk factor. So today I would like to share with you one of my research project started by me during my training in UK, but completed by my student, okay, entitled Profiling of MicroRNA Landscape Within Human Vascular Smooth Muscle Cells and Exosome During Replicative Senescence. Okay, so I think this gray bar here is blocking your view, but is at the same time it blocked my view, so I'm not sure how to remove this, but okay, I will try my best, okay, to deliver. Okay, so first of all, I would like to talk about microRNA. So I'm not sure how many of you are familiar with microRNA. So microRNA is a small non-coding RNA of around 19 to 25 base pair. So in the past 10 to 20 decades, this microRNA research have been the spotlight, okay, in the research world, okay. And in terms of the microRNA biogenesis, there are actually 5 steps, okay. So you have the canonical microRNA biogenesis, the non-canonical microRNA biogenesis. The only difference between these two pathways is it involves a DROSHA and DGCR8, the nuclear processing. Okay, after this step, okay, the premature microRNA will be exported out from the nucleus and then enter the cytosol, where it will be then the mere RIS assembly will form. Okay, and then finally this microRNA will bind to the 3' UTR of mRNA, and then to elicit the function. So basically, it's either it leads to mRNA degradation or it leads to translational repression. Okay, so that is the classical function of microRNA in the cells. But the microRNA can also be packaged in the exosome, okay, and then being exocytosed outside the cells, so that this microRNA get carried away in the exosome and exported to other places to basically do the same function. Okay, the function is again mRNA degradation or post-translational repression. So microRNA is basically a very important and play a very important role in transcriptional regulation, okay, of a gene, and therefore it can change the cellular behavior as well as the cellular phenotype. Okay, so next I would like to talk a little bit on ischemic heart disease and ischemic heart disease created by atherosclerosis. So as shown in this diagram here, there is a very healthy blood vessel. And in this diagram, you can clearly see that there is atherosclerosis or atherosclerotic plaque build-up here. So just a little bit on the introduction on the atherosclerosis process. So for the atherosclerosis to happen, there are few prerequisites. So for example, you need to have a very high level of cholesterol and also followed by endothelial injury. And also you need to have oxidative stress. Okay, so these are some of the prerequisites for atherosclerosis development. So say for example, when all these factors are in place, so we have cholesterol that will invade into the subendothelial space, okay, and then become oxidized to become oxidized LDL. The patrolling monocytes will chase after the oxidized LDL, and then they will swallow the oxidized LDL and become foam cells. So these foam cells will secrete a lot of cytokines as well as signaling molecule, leading to the migration of vascular smooth muscle cell. So basically, this atherosclerotic plaque is covered by human vascular smooth muscle cell. So this is human vascular smooth muscle cell. So atherosclerosis process is a chronic process. Okay, it's a chronic process. Probably it needs 20 to 30 years to develop any obvious symptoms. Okay. So towards the later stage, or the patient will start to feel like heart palpitation, chest pain, shortness of breath, dizziness, or even loss of consciousness. Okay. And then when this occurs, usually the atherosclerotic plaque is already quite huge. And then due to the turbulence in flow, as well as very high blood pressure in this region, it can usually lead to plaque rupture. So when plaque rupture, okay, so the tissue factor will be exposed to the blood, and therefore the blood clot will happen inside the lumen of the blood vessel, which is, we call it as thrombosis. So thrombosis happen in an acute manner and usually is fatal. Okay, so thrombosis is quite dangerous. So in the past, when I was in UK, so my mentor, he is a physician. So in his laboratory, he has two teams. So one team is clinician. So they try to identify thousands and thousands of echocardiography images and trying to find a certain trait from the images, to identify whether, the patient will have an unstable plaque. Okay, so this is a stable versus unstable plaque. Okay, so he also has another team of scientists like me. So we are trying to find a biomarker to basically predict whether the plaque is stable or unstable. So if it's unstable plaque, so probably the clinician will need to quickly schedule for operation to either put a stents or go through the bypass surgery. So that is basically what happened when I was in UK. So then I come up with this hypothesis for my research work. So I hypothesized that vascular smooth muscle cell senescence mean aging, yeah, very old. Okay, vascular smooth muscle cell senescence lead to plaque instability. So, okay, and therefore lead to the thrombosis process. So should you still remember, just now I did mention that, the cell layer that covered the foam cells, these are human vascular smooth muscle cell, okay? Okay, so this is my hypothesis. And then what were the problem statement? So at that time, probably close to more than 10 years ago, the signature microRNA expression profile of human vascular smooth muscle cells and their exosome during a replicative senescence was still unknown, and thus lead to the objective of our study. So our study, we are trying to perform characterization and profiling of microRNA from exosome, either inside the cell. So these are the microRNA from exosome, either inside the cell or secreted outside the cells, okay, during replicative senescence. Okay, so this so now I'm going to talk about the method. Since it involves human vascular smooth muscle cells, so we need to apply clinical ethics from from Ministry of Health Malaysia, and also from the ethics committee from the university. And then after that, we need to really characterize our human vascular smooth muscle cells. They are really old, and they are really underwent the replicative senescence process. Basically, it's just the aging process, okay? They are really old. And then finally, we also need to confirm our technique to isolate exosome from human vascular smooth muscle cells were correctly performed, okay? And then we will sequence the microRNA and then analyze the data, and then to validate the differentially expressed microRNA. Okay, so again, as shown here, we have a young human vascular smooth muscle cell and the old human vascular smooth muscle cells. So first of all, before our experiment, we need to ensure they are really underwent replicative senescence. So we stain the cell using SA beta gal staining at pH 6. So those cells that are really old, they will stain positive. Okay, they will stain positive for this blue color. And then we also isolate the total RNA, and then we do a qPCR to look at the senescence associated gene marker. For example, P21, P27, and P16. Okay. However, these two methods are generic methods to look at senescence. So when we talk about senescence, we actually have two types of senescence. The first one is stress-induced premature senescence. So which means that, for example, you treat a cell using a very harsh condition or harsh chemical. So, you basically accelerate the aging process. So that's why we call it as stress-induced premature senescence. For replicative senescence, it's just a normal senescence process. So when cell divide, because it has a limited cycle, so then the cells become old. Okay, so that is the normal senescence. So in our study, we only look at replicative senescence. So for SA beta gal staining as well as the SA gene marker, these are the generic senescence marker for both stress-induced premature senescence as well as replicative senescence. But for the telomere length measurement, this is the assay that is specific for replicative senescence. So just for information, telomere is just a cap on the chromosome. So every time when cell divide, the telomere length will become shorter and shorter, okay? And therefore, every cell has a limited lifespan, yeah, because the telomere length is already shortened to a certain level, okay? So then we also extracted the gDNA and performed a telomere length measurement. And also we perform ultracentrifugation to collect exosome, either from the cells or the exosome secreted outside the cells. So the ultracentrifugation process usually involves 16 hours at a different speed. And then after that, we will check whether our exosome isolation technique is okay by using Western blot. So we check one of the exosome surface marker, which is TSG101, and also we view the exosome using transmission electron microscopy. Okay, after we have confirmed the cells are all, and then we confirm our exosome isolation techniques were all correct, then we will isolate total RNA, okay, from exosome from inside the cell as well as exosome excreted outside. So we will check the RNA concentration using qubit, and then after that check the RNA integrity using bioanalyzer. And then we are using Illumina TrueSeq library prep, okay, to prepare our CDN library. So these were the steps involved. For example, we will ligate the adapter, amplify the library, and then purify the cDNA construct, and then we will check the library integrity, and then also we will normalize the library concentration because we have multiple samples. And then now everything was ready, so we will send for sequencing. Okay. After we've gotten the sequencing data, so we'll perform primary, secondary, as well as tertiary analysis. So primary analysis basically just for us to look at the sequence reads as well as the quality score. And then for the secondary analysis, we will trim the adapter sequence, and then we'll do some alignment. So tertiary analysis, that will be the one that give us the functional data in which we require for our study. So in detail, for the tertiary analysis, we are looking at the differentially expressed microRNA. So first of all, we need to predict, what are the mRNA targets, okay, for the differentially expressed microRNA. So in this way, we use MIRWalk and target scan to analyze our sequence, and also we look at microRNA and mRNA interaction network. We also use gene ontology as well as pathway analysis and protein-protein interaction to basically analyze our sequencing data. Also, we validate the differentially expressed microRNA using qPCR. Okay, so now let's look at the data that we obtain. So first of all, we need to confirm the human vascular smooth muscle cells underwent replicative senescence. So just now, as I mentioned, you can see these were the young cells, and these were the old cells. So the old cell will have a very, very different morphology. You can see they are flat, and of course we have less number, okay, because they underwent cell cycle arrest. So you can see they basically stain positive for SA beta gal staining. So when we do the qPCR to check of senescence marker, you can see P16, P21, and P27's level were up. Then when we measure the telomere length, so for the senescent cells, the telomere were basically shorter. So indicating that we already have a population of human vascular smooth muscle cells that underwent replicative senescence. The next study, we confirm that our exosome isolation technique, either from inside the cell or secreted outside, were correct. So we use transmission electron microscopy to view the exosome isolated. So as you can see, the exosome isolated from O cells, they are more clustered, okay? And also interestingly enough, when we probe for surface marker for exosome, the exosomal, the extracellular exosome, we can see an increase in about 20 kilodalton. So indicating that that is a novel post-translational modification that is essential for the secretion of the exosome from the cells to outside, and which has never been reported. So this is a new research question in which now we are actually following the leads. So in conclusion, we have successfully isolated exosome from our culture media and also from inside the cells. Okay, so finally, after the sequencing, so this will be our heat map. Okay, so this is the microRNA profile from exosome inside the cell, and this is the microRNA profile from exosome outside the cells. So altogether, we obtained more than 127 million reads, and out of which only 3,806 were mature microRNA. And when we do a deeper analysis, only 73 that were differentially expressed. But, you know, due to funding constraint and things like that, so we cannot validate all these 73 microRNA. So then we use FDR to adjust we use the FDR adjustment, okay, to find those microRNA that were really very significantly expressed. So as you can see here, for intracellular microRNA, so we have identified 2 microRNA that were downregulated and 6 microRNA that were upregulated. So for exosomal microRNA, we identified 2 microRNA that were downregulated and 1 microRNA that was upregulated. Okay, so this was after the FDR adjustment. So we identified in total like 11 microRNA in which, you know, we will focus in our study. Okay, so after we have identified the microRNA that we can use in the later on in the clinical settings, but we need to first to validate their expression first, okay, using qPCR. So as shown here, so this diagram show the intracellular upregulated microRNA. So you can see their level all up, and these are the downregulated intracellular microRNA. So these 2, but for exosomal microRNA, these 2 were the downregulated one. As for the upregulated microRNA, we were not able to validate the expression because we were not able to design the primer because the primer is rich in GC content. So we were not able to design the primer. Okay. So in conclusion, for novel upregulated intracellular and exosomal microRNA in human vascular smooth muscle cell, okay, so for intracellular microRNA pool, so we have identified 4 microRNA, MIR664A3P, MIR664A5P, MIR664B3P, as well as MIR4485 3P. So basically, they involve in protein phosphorylation, subcellular localization, and intracellular transport. As for exosomal microRNA that were upregulated, you know, we identified MIR7708. So when we did all the analysis, we found out that it actually involved in intercellular communication. Okay, so with all this data, it makes us even more puzzled, okay, because it's too broad, okay, the function. However, when we look at the downregulated intracellular as well as exosomal microRNA in human vascular smooth muscle cells, okay, for intracellular microRNA, the downregulated one were MIR1555P and 20A5P. For exosomal, again, we saw the similar microRNA 1555P as well as 146B5P. Then when we did the analysis, we found out that these microRNA were basically involved in increased oxidative stress, involved in cytoskeletal reorganization. So just now, as I mentioned, in aging, oxidative stress will increase. And also you can see from the young cell and old cells, their morphology is very different. So we can see cytoskeletal reorganization and also cell cycle arrest. Okay, both the downregulated microRNA. So we can see cell cycle arrest come in as well as reduce inflammation. Okay, so we were excited because this downregulated microRNA basically they involve in the pathway that give us the hallmark of cellular senescence or cellular aging. And importantly, we consistently saw MIR1555P happen either inside the cells or in the microRNA inside the cell or outside the cells. So when we basically look a little bit deeper, research literature online to look a little bit deeper on MIR155, we found out that this MIR155 basically involved in atherosclerosis, okay, the foam cells formation, and also MIR155 enhances atherosclerosis process, and it makes the plaque become non-stable, okay, in the animal model. And also, if we isolate MIR155 and we transfer MIR155 from smooth muscle cell to endothelial cells, and the endothelial cells will become injured and then they promote atherosclerosis. So all these indication showing that MIR155 is very important in promoting vascular health. So if the level of MIR155 is low, it involves in development of atherosclerosis as well as it makes the atherosclerotic plaque not stable. And also, we can see in this very recent literature, like just 4 years ago, MIR155 is implicated in hypertension. So in our study, we identified MIR155 also as a very potent, potential microRNA. And this interesting microRNA basically link atherosclerosis development, hypertension, and vascular aging together. So in summary, so we have already identified sets of microRNA from inside the cell, from exosome, from the human vascular smooth muscle cell, inside as well as outside. Okay, so the downregulated microRNA involved in cell cycle oxidative stress as well as cytoskeletal reorganization. Okay, and then the upregulated one in the intracellular, we found a lot with unknown function. As for the exosomal microRNA, the downregulated one involved in proliferation and inflammation, but the upregulated microRNA, also, these are the novel microRNA with unknown function. So, basically, we are successful in identified sets of microRNA, which in the cellular senescence, which might be very important for us to look at vascular aging as well as vascular related diseases. Okay, in conclusion, so what is new about our study? So for information, our study, we were the first group to profile intracellular and exosomal microRNA in human vascular smooth muscle cells during replicative senescence. So during our research, right, we actually find out that when cells, when they are aging, when they are senesced, so the exosome we isolate from aged cell, they tend to cluster together. So we do not know why. And also, we identify a novel post-translational modification that is essential for the excretion of exosome from inside the cells to outside the cells. Okay. So, and this marker is TSG 101. Also, we identified novel upregulated intracellular and exosomal microRNA. So basically, we also discussed about the potential of MIR-1555P as a biomarker of human vascular smooth muscle cells during replicative senescence. So what is the clinical implication? So we basically can use sets of microRNA to predict hypertension or to predict atherosclerosis development, okay? And then we can also use this microRNA as our drug target to basically cure some of the vascular related diseases. Okay, so now we come back and we revisit the slides that I proposed initially, the hypothesis. So, you know, we come back to the hypothesis. So the initial hypothesis was the plaque rupture. Yeah, the plaque become unstable, and therefore we need to help the clinician in terms of the decision making, because we have a lot of patients waiting for cardiothoracic surgery, but we cannot handle everybody. So we need to ensure those patients that are really on emergency, so they are put in front first. So probably the sets of biomarker that we identify, especially like MIR-155, can potentially, you know, help clinician in the decision making in order to save life. So currently, we are using our sets of microRNA that we identified from cell culture. So now we are actually validate them in the clinical settings. Okay, so finally, I would like to acknowledge my mentor, Professor William Chillian from Northeast Ohio Medical University, United States. I would like to thank my collaborator, Dr. Samso Muhammad Zain from University of Malaya, Dr. Loh Teck Yew from UMBI, Dr. Hamza from Hospital Sedang, Dr. Muhammad Fauzi Daud from University Kuala Lumpur, the funder, Ministry of Higher Education, for awarding me the grant to carry out the research. I would like to also thank the University of Nottingham to allow me to use the research facilities as well as a research space to carry out our research, and friends, colleagues, and family. And of course, this work was mainly done by my PhD student, Dr. Nguyen. So she is actually Vietnamese. So then she traveled all the way to Malaysia and did the PhD with me. Okay, so she is a very talented young scientist. Okay, so finally, this slide shows a very beautiful campus, the University of Nottingham, Malaysia. So this is, I would say, University of Nottingham, Malaysia has one of the most, I cannot say the most beautiful, but one of the most beautiful campus in Malaysia. So hopefully one day, you know, some of you can come, if you come to Malaysia, please come to visit us. So we basically located a little bit south from Kuala Lumpur. Okay, so these were my lab members before COVID. Okay, with that, I thank you for your attention.
Okay, thank you very much, Prof. Pung, for a very insightful presentation. I assume there would be a lot of questions from the participants, because I see some of them are already raising their hands currently. So we would now proceed to the 20 minutes Q&A session. I would like to ask for Mr. Purbianto to unmute yourself and then directly ask questions to Professor Pung.
Oke, thank you moderator. Untuk moderator nanti mohon diterjemahkan ke bahasa Inggrisnya ya.
Boleh,
Ya.
boleh, silakan.
Oke, baik. Saya ini penelitian yang cukup menarik ya. Sebenarnya ada kaitannya dengan apa yang saya sampaikan tadi. Artinya bahwa pada pasien-pasien yang hipertensi, kemudian dia beresiko untuk terjadi penumpukan plak, ya, kemudian plak yang akan menyebabkan terjadinya cardiac arrest. Gitu ya. Nah sekarang kan yang lebih bagus bagaimana kita mencegah terjadinya plak itu, gitu kan? Nah sekarang kan artinya bahwa plak itu kan baru bisa dilihat kalau sudah dilakukan kateterisasi kan ya. Jadi dengan kateterisasi bisa lihat plak. Nah sekarang bagaimana ataukah mungkin Prof. Fung ini mungkin sudah pernah mengembangkan bagaimana mendeteksi secara dini dengan biomarker yang ada di dalam pembuluh darah untuk mendeteksi dini bahwa ini kemungkinan akan timbul plak akibat hipertensi itu gitu. Nah, artinya dengan kalau kita bisa mendeteksi biomarker itu, nah kan mungkin bisa menjadi deteksi dini gitu supaya tidak terbentuknya plak yang lebih besar lagi. Nah, gitu mungkin adakah biomarker yang bisa mungkin dideteksi secara dini untuk supaya ini sudah mulai terjadi plak ini. Bagaimana kita mencegahnya untuk supaya tidak jadi plak yang lebih besar lagi. Mungkin itu, terima kasih. Prof.
Baik, Pak Purbianto, saya izin untuk mentranslate-nya ke Profesor Pung. So, Professor Pung, Mr. Purbianto would like to inquire about the availability of any biomarkers such as circulating microRNAs to predict or prevent the plague, because we all know that the plague could lead to cardiac injury such as cardiac arrest. So it would be better for us to, at least like by profiling the microRNAs on some patients, we would have this grasp of prediction whether he or she would develop plague in the future. Is there any other possibilities to predict?
Of course, we can use microRNA to predict. We can use microRNA to predict from I would say at this stage, I think we were the first one to come up with these sets of microRNA. This come from my previous work when I was in Cambridge University. Okay, so as I did mention, if you want to predict whether the plaque is stable or unstable, so my previous mentor, my previous laboratory, they also analyzed the echocardiography images. They are trying to find out, what are the traits leading to non-stable plaque. But in terms of the biomarker from microRNA, I think our group was the first one to come up with these sets of microRNA. And now we are currently trying to validate this in the clinical settings. We try to work with a cardiothoracic surgeon. Yeah.
I see.
Yeah.
Probably we could, other than microRNAs, currently there are a lot of studies as well, try to do the polygenic risk scores to see such gene polymorphisms that could interact each other, gene and gene interactions, to predict the polygenic risk score for the incidence of plague. Some people are trying to conduct that as well. What about your opinion on that?
Okay, so I would think because I think we have a precision medicine consortium, Southeast Asia. So we know we have participants from Indonesia, like Malaysia, Thailand, Philippines, even Brunei. We all participate in this precision medicine consortium. But I think for Malaysia, I would like to say we are quite ashamed because we are very, very behind. Okay, we are very, very behind. So in this Southeast Asia region, the most advanced country in Southeast Asia region is Thailand. Even Singapore also very lag behind. So in Thailand, they already have the population study. They already have population study in which they collect a lot of data, and then they try to analyze their own population. Their polygenic risk score, PRS, as well as PGX, the pharmacogenomics gene. So they already have this data bank 10 years ago. So after 10 years, I mean, for other countries, even the most developed country, Singapore, we are also all lag behind. So Thailand is our big brother of all the Southeast Asia region. So as in Malaysia, we are trying to drive the precision medicine in the university to raise awareness, to try to incorporate this in the education in our curriculum. Okay, but as I said, we are lag behind. So we need to learn from Thailand.
Yeah, true. Because there are a lot of studies try to combine not just the biomarkers, but also with other risk factors such as BMI or blood pressure and other
Yeah.
things.
Yeah, but this one involve a population study in which you need a lot of genetics information from the population. Then you do sequencing and involve a lot of money as well. So it only can funded by the government.
Oke, jadi untuk jawaban dari Prof. Pung, mungkin Pak Purbianto juga sudah bisa menangkapnya ya Pak Pur. Memang saat ini masih belum ada biomarkers yang khusus untuk plak. Tapi untuk hipertensi sendiri atau tadi yang sudah dijabarkan oleh Prof. Pung, beliau adalah kelompok peneliti pertama yang menemukan bahwa ada sepaket microRNAs yang bisa digunakan untuk memprediksi mengenai kejadian hipertensi atau aterosklerosis itu sendiri.
But we haven't validated this in clinical settings.
Yeah, that's another homework for all the researchers.
We haven't validated that. Yeah, we in the process of validating it.
I wish you all the best for the research team. Okay, let's move on to the next question from Ibu Heni. Ibu Heni mungkin bisa langsung mengutarakan pertanyaannya, Ibu Heni, atau bisa menyalakan kameranya terlebih dahulu.
Yeah, I'm so sorry I could not on cam because my signal is not stable. So Professor Fung, I would like to ask to you about the tool you said before that you have echo. Is that right? For
echocardiography. Echocardiography.
Yeah.
I don't have. I have this when I was in UK, when I was in Cambridge. So because my mentor was a clinician. Yeah, so he has this tool, but when I came back to Malaysia, I don't have this. So I can only work on my expertise, trying to look at biomarkers.
Oh, I see. I'm so sorry. It's mean your project have result that the situation for the cardiovascular in coronary disease could not prevent but detect. Is that right, or I'm wrong for your preparation?
So basically, we are I think in the past, right, my mentor laboratory was trying to identify the stable versus non-stable plaque. So if the plaque is non-stable, so the physician will need to schedule for operation, right? Because when the plaque rupture, then it becomes thrombosis. That is very dangerous for the patient. But the patient is a lot in the hospital, so we need to find ways to help the clinician in terms of decision making. Yeah, like which patient should come first. So we will couple using echocardiography.
Yeah.
So, but these were mainly done by clinicians. So they will echo, then they will collect the photo, then they will compare and things like that. So, and then another group, we will try to find biomarkers, like biomarkers that are very dangerous, like from patient that they already have severe atherosclerosis and with unstable plaque, or the patient before going to operation. So we will take the blood and things like that, and we do analysis from there.
Oh, I see. Thanks a lot, Professor Fung. Nice to hear that. Thank you, Miss Zizki.
Yeah, I hope I can have collaborations from Indonesia. So, so hopefully we can
like to hear that.
And you can come to Nottingham, Malaysia.
Okay.
Amen.
Thank you.
I'm very agree, yes.
Is there any other questions from the audience? I would like to highlight that it is really interesting that there is a possibility of post-translational modification for the exosome. So we would probably see the possibility of epigenetic modifications for some certain cases probably. Would it be a possibility to have epigenetic playing a part in your finding previously that you mentioned?
Of course, I think I think now with the role of precision medicine. So precision medicines, we are looking at our genetics. Yeah, we are looking at our genetics and our genome. But our genetics, we cannot change anything because we are born like that. We can't change anything. Probably only, but you know, the epigenetics also is part of the precision medicine. Epigenetics means on top of genetics. So it's even above the genetics. So this on top of genetics factors is with the environment and as well with the person itself. So epigenetics, you know, you can basically change the outcome of the genetics by controlling whether a certain gene or certain protein will be upregulated, will be expressed, or it will be repressed. So if genetic play 30% of the role to determine the disease outcome, but the epigenetic probably you have 70%. Yeah, so basically you can change the disease outcome, especially in non-communicable diseases. So microRNA is one of the epigenetic changes. Of course, methylation, acetylation, you know, these are also expect in the epigenetics.
Okay,
Yeah,
sure.
so we can basically change the disease outcome.
Okay. Based on your finding, you said that the microRNA 155P is one of the proposed biomarkers for this situation. I would like to confirm that the microRNA 155P is the one that is expressed in the human vascular muscle.
Is expressed and is required, I think, based on our study, is required to maintain the vascular health. So the level is high, so the vasculature will be healthy and happy. So if the level is low, okay, so it's basically involved in, you can see atherosclerosis, vascular aging, like in our study. And also from other people's study, you can see atherosclerosis as well as hypertension. So indicating that this smear is important. But usually when we design biomarker, we will not only use like one microRNA. We will use sets of microRNA to basically predict. Yeah, that's what I personally feel.
Would it be different if you try to look at the one that is circulating, like circulating mRNA? Would it be give different result? Would it give different result?
I think so. I think so. I think there are a lot of people, when you look at the publication, you know, they just collect the bloods and then they just isolate. But I think it's a fast way as for diagnostic, because you do not need to separate whether, you know, this is from exosome or for what. It is a fast way, but I think for us, we try to do a little bit a cleaner version. You know, we try to ensure it comes from this, and then we try to, yeah. Of course, you can come just to isolate microRNA, and is easier also.
Yeah, because currently there are more studies trying to look at the circulating instead of the one.
But if you look at the circulating microRNA, so exosome also a subset of circulating microRNA.
Yeah. Okay. Is there any other questions from the audience? But I think also the time is limited, so we would like to thank you once again to Professor Pung for your time and all the knowledge and experience shared to us. Like you say, who knows, in the future, one of us would probably come visit Nottingham, and probably you would come visit us in Indonesia. And then we would have such a nice collaboration and research in terms of your expertise of fields. We all surely walking away from this webinar with new updated knowledge and valuable insights. Last but not least, thank you to you all, the audience, who have actively participated in this session. On behalf of the committee, we thank you all and we wish you a fruitful experience from this webinar today. Probably before we end this session, we would like to have a quick photo shoot together. To all the audience, would you mind to turn on your camera, and then we would have a photo session. Kepada para peserta, dimohonkan untuk menyalakan kameranya. Kita akan mengadakan foto bersama dengan para pembicara. Oke. Masih dinantikan kepada para peserta lainnya. Ini mohon Aba untuk yang mengambil foto, apakah dari panitia atau saya akan membantu untuk mengambil fotonya juga? Oke. in the count of 1, 2, 3. 1, 2, 3. Okay, terima kasih. Thank you very much once again, Professor Pung, for coming today.
Thank you, thank you. So we will have a conference with our partner collaborating university from Thailand next year, August 2023. So I'm not sure, whether we can also, invite you people to come over to Malaysia next year, August. So we will discuss this in detail, okay?
Yeah, thank you.
Okay.
Today, there is also a genomic and genetic conference counseling in Malaysia, the Hospital Pakar Kanak-Kanak, UKM.
Oh, okay.
There's also a conference there.
Oh, okay.
I actually joined it online, so I have 2 devices here.
Oh,
Then
you must be very
next year, August. Thank you very much once again. Nice to meet you.
Thank you. Bye-bye. Bye-bye.
Okay, thank you, Ms. Lupa.
Bye-bye.
Terima kasih kepada para peserta. Saya akan kembalikan acara ke M. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor Yu Fan Hu. Ladies and gentlemen, now we are moving the next speaker of the webinar. The next will be Professor Irmanida Batubara, who will deliver the topic of the use of native Indonesian herbal medicines for prevention of non-communicable diseases. The second session will be moderated by Ms. Jiske Mariska again. To moderator and speaker, please take your time.
Terima kasih kepada MC. Untuk selanjutnya, sesi berikutnya, ini adalah pembicara ketiga kita, Profesor Irmanida Batubara. Mohon kepada panitia untuk menampilkan CV-nya. Profesor Irmanida Batubara merupakan salah satu peneliti yang berasalkan institusi dari Pusat Studi Biofarmaka Tropika LPPN IPB, Departemen Kimia FMIPA IPB, Kampus IPB Darmaga Bogor. Beliau merupakan seorang peneliti dengan banyak sekali publikasi dan juga pengalaman. Bisa dilihat di CV beliau dengan High Index Scopus dan Google Scholar. Oke, kita lihat juga masih banyak sekali riwayat pendidikan beliau, mulai dari S1, S2, dan juga S3 di Gifu University, Jepang, dan juga pengalaman penelitian dalam 5 tahun terakhir yang tidak bisa saya sebutkan satu persatu. Tanpa berpanjang waktu, untuk sesi berikutnya, saya izin untuk meng-offer ke Bapak Ahmad Yani untuk memandu acara presentasi dari Prof. Irmanida Batubara. Terima kasih.
All right. Thank you, Ms. Ziske, for joining us today by moderating the first session with us. And due to other circumstances, Ms. Ziske will probably hand over to me, and this second session will be delivered by Professor Irmanida Batubara from IPB University, will be moderated by me. As introduced by Ms. Ziske before, we may be straightforward to invite our second speaker, to Professor Irmanida Batubara. Are you ready, Prof?
Yes, okay, I'm okay.
All right, thank you very much. So probably we directly hand this session over to Professor Irmanida Batubara. Professor Irmanida Batubara, the time is yours.
Okay, thank you. Pak Made and then Mbak Ziske previously. Since the participant is Indonesian, do you want me to talk in Indonesia or still in English? Can you hear my voice or no? Hello, is it clear, my voice?
Clear, Prof.
Oh, clear, okay. Okay, thank you very much. So, thank you for the committee to invite me to give talk about the use of native Indonesian herbal medicine for prevention of NCD. So, this is the agenda that I prepare. The first is about IPB University and also Tropical Biopharmaca Research Center. And then the second is about NCD, but I think it's already explained clearly by Ms. Fung previously. So I will omit this part maybe. And then after that, to native Indonesian herbal medicine and diabetic, also body weight and inhalation of native Indonesian herbal medicine, and finally closing statement. So I will move for the first agenda for IPB University. So this is our university. If we see in Indonesia, we have Java Island, and then Bogor is located about 60 kilometers from Jakarta. And we have several campus in Bogor. This part is in Darmaga campus, but this one in Baranang Siang. Our place is near to the Presidential Palace and also Bogor Botanical Garden. Please come to Bogor if you have time and you can visit us. So IPB or Institute Pertanian Bogor, but mostly translated into Bogor Agricultural University. And now we have the name, specific name for our university become IPB University. This university is one of the major higher education in Indonesia. We has a good reputation and also play an important role in the national development and higher education in Indonesia. Since all the university has three main responsibilities in Indonesia, namely education, research and development, and as well as community services. So IPB has 10 faculty and also 24 research center. One of the research center is our research center. The name is Tropical Biopharmaca Research Center. So why IPB has Biopharmaca Research Center? As we know together that Indonesia is mega diversity countries. We have a lot of things from nature, plant, animals, microbe. Also, we can find a lot of type in Indonesia of this species. So we define the name of biopharmaca. So biopharmaca is plant, animals, and microbe that have potential properties as drug, health food, functional food, and also nutraceutical for human, animals, and also plants. So traditionally, biopharmaca in Indonesia name is jamu that already consumed since ancient time. Yeah. So now we want to give more scientific data for our traditional knowledge. So Tropical Biopharmaca Research Center is the name of the center. The center is an organization that belongs to Institute of Research and Community Services, or in Indonesia we call Lembaga Penelitian and Pengabdian pada Masyarakat. Ya. This research center is a networking institution and coordinating research on biopharmaca related materials. We also working on biopharmaca development, natural resources, conservation, proposing the bioprospective policy and also regulation. We have multidisciplinary expertise, especially for exploration, conservation, cultivation, extraction, chemical structure analysis, standardization, and efficacy study of natural resources. Here are the founder of our research center. Maybe Pak Ahmad Yani still remember Prof. Latifah Kadarusman, the late Prof. Latifah Kadarusman. We also have Prof. Tony Ungerer, also Prof. Dondin, and Prof. Suminar, also Prof. Dudung Darusman. Yeah, our research mostly coverage from exploration of Indonesian medicinal plant or sometimes microbe, also from animals. Yeah, we did domestication, cultivation, and also to produce good agricultural practices. From the exploration, we screen the biological activity with using bioassay, and then we found a specific plant or specific animal, specific microbe that we can develop. And then using biomolecular analysis and phytochemical profiling, hopefully we can get the standardized raw material, especially to make a formulation. Yeah, we formulate the herbal medicine using biomolecular and bioinformatic approaches, and we hope we can get the standardized Indonesian natural product medicinal product. So this is all the research, and I will talk mostly related with this coverage. Yeah, now we move to the second part for non-communicable diseases, as mentioned already from Prof. Hung, so I will not mention in detail anymore. But I will inform you that my research focus on, my talk is focused now on diabetic, yeah. So this is the information about Indonesia. As we know already, we have a largest archipelago in the world. We have a very big land, also ocean, and population as well. And now we are on the third youngest in East Asia for the population. But we hope all the population are in good health, not in bad health. Yeah. So the current situation, this is the last report that I can found. The main causes of death in old age in Indonesia, the same like in Malaysia, is stroke, coronary heart disease, and also diabetics. Yeah, that the one that I will discuss on this at this time. So diabetics, and with this complication, tuberculosis as well as COPD. Yeah. And sorry, so okay. The other is for risk factor of NCD. I will focus on the obesity. Yeah. So in the last talk, I will inform or give little bit our research related to inhalation and also obesity. So all for risk factor as well as the diseases, we use Indonesian native medicine. So now let's move to diabetics. Yeah. So for diabetics, we search all Indonesian native resources, because Indonesia have different natural resources. In different place, we have different natural resources. And also in different place, we have different ethnic groups. Yeah. Also different environmental condition. It makes we have a lot of formula for diseases, specific diseases. So we need to give a scientific information for our traditional knowledge. Yeah, now for diabetics, I will move first with the research related to bioinformatics. Yeah, sorry, this is still in Indonesia. So search, we first we collect all Indonesian plan, yeah, and then search the information from Knapsak database. After that, from Knapsak database, we can find some compound that already reported existing in this plant. From the compound, we search in PubChem, yeah. So how about this compound related to the protein target? Yeah. So this is one part. And on the other hand, we check also the same for the protein target. This is the A, this is the B. And then after that, we check related to the disease, especially for diabetic. From this research, we check the Knapsak, because in Knapsak, we can find jamu here. And from jamu, we can find the compound that existing in the plant already reported by the other researcher. So from there, we put in IJAH analytics. Yeah, IJAH, you can check in this website. So when we search for diabetics here, Actually, we can found about 532 plants and 185 compounds that can be as an anti-diabetic. Yeah. So from this, we search which one is most prospective, which one is not most prospective, based on the computer analysis. So this is the way using graph mining, yeah, to find what is the prospective plant. So from this research, we can found about 1,467 active compounds. That this compound we can found in 460 plants. Yeah. Interestingly, this research the results say that Mangifera indica, if everybody knows, Mangifera indica is mango. Mango is the most prospective for anti-diabetics. But traditional knowledge, we don't know how Mangifera indica become an anti-diabetics. Yeah. The other is Phaseolus vulgaris, buncis, and also Delima, yeah, Punica granatum. So from this data, because they said the result said mango is the most prospective for anti-diabetics, we make a research with the student, yeah, for for alpha glucosidase inhibitors and alpha amylase inhibitor, because we use this assay to stop the conversion from carbohydrate become glucose. Yeah, we did not check the inhibit glucose digestibility and also absorption, but we check only the two here: alpha glucose inhibitor or alpha amylase inhibitor. So this is the result. Since Indonesia is very rich, yeah, with biodiversity, at that time we only select 3 types of mango in Indonesia: Indramayu, Arum Manis, Manalagi, and also Budiraja. Yeah, so we collect this plant, and the question is, the active component, is it in the peel, is it in the flesh, or in the seed? So we don't know in what part is the active component. And the student try to tidak tampil di layar Zoom PPT-nya. Apakah yang lain tidak melihat layar Zoom-nya?
We can see
okay,
your
good. Thank you.
presentations, Professor.
Sorry. Because in the chat box, somebody said cannot see the PPT. Okay, I will continue. Yeah, thank you. Yeah, here we check from the peel or kulit mangga, and the endosperm, the inside seed, and also exosperm, and then the flesh. Yeah, and several data we can found here. Interestingly, when we consume the Manalagi not consume, I mean the extract of Manalagi endosperm ethyl acetate is the most prospective for anti-diabetics from this result. Yeah, because it has lowest IC50 for alpha glucose inhibitor. Sorry. Okay. And then after that, traditional knowledge in Indonesia said the snack fruit, yeah, snack fruit peel also has potency as anti-diabetics. So we collect several type of snack fruit in Indonesia. We have salak pondo, salak cililin, salak malaka, salak manojaya, salak pondo mekarsari, salak condet, and also salak mawar. Yeah, and then from here we separate between the skin fruits and also flesh extract. And then the results showed that, this is the information, the place that we collect the snack fruit, yeah, from different place, and also from Mekarsari, Taman Buah Mekarsari in Jonggol, yeah. The result we can found here that they have several phenolic and also flavonoid content, and the most prospective is this one that can inhibit alpha glucosidase activity only in 17.9 microgram per milliliter. This we can found in skin fruit of Manonjaya salak or Manonjaya snack fruits. So interesting data that actually we can develop this product, skin fruit to be a product, but still not continue yet until now. This is the information, the activity we check by TLC autogram with alpha glucosidase. Yeah, we can found selected spot that if we continue, we can collect the compound and then estimate what is the compound responsible for the alpha glucosidase inhibitor. The other information is related to yogurt because we maybe like to consume yogurt, but this yogurt is added by Rosella extract, supplemented with Rosella extract. And we check the changes of activity during storage. Yeah, this is different duration of storage. So we can found that when we storage it more longer, the activity become decrease. Yeah, this is the information from the result of our research. The other is from sweet potato. Yeah, but this is the leaf of purple sweet potato. So the result also have reported that we have a potency for anti-alpha glucosidase inhibitor, especially for alkaloid fraction, not for anthocyanin fraction. But the most prospective is the alkaloid fraction that can inhibit about 61.9% in the concentration of 3%. Still, we have for alpha glucosidase inhibition. Yeah, this one from red kidney bean. Yeah, from red kidney bean, and then we already make the fraction of the red kidney bean, and we check the structure related to quercetin glucoside. Yeah, here, and then we find a different IC50 value. Here, after this. Actually, almost all fractions are active, but only A fraction is the most active one compared to other fraction. The other, not a red kidney bean, we have also information about jack bean. The jack bean, here we check the effect on thermal stability. So when we give heat on the jack bean, what is the activity of alpha glucosidase inhibition? This is also related to the compound. Yeah, the compound here is chimeferol anisoyl chimeferol glucoside. So different glucoside give different activity, and we can found different amount of glucoside in each thermal stability, different temperature. I mean, yeah, this is the information. So we collected compound 1, compound 4A, 4B, 4C, 5A, 5B, and also 5C. Yeah, this is the glucoside, different glucoside, R1 and also R2. This is R2 and this one is R1. Yeah, the activity also different. We only found only check the inhibition activity, not IC50, due to limitation of the number of compound isolated from these samples. The others is from vegetable and also spices. Yeah, so we try to select different number, different vegetable and also spices in Indonesia. This is salam, daun salam, and then daun jambu mete, all here, torbangun, and then also ginseng leaf, daun pandan leaf. Yeah, and after that we try to focus on salam leaf or Syzygium polyanthum. Yeah, this one, Syzygium polyanthum. We use the metabolomic approaches and also in silico approaches for this activity. Yeah, from the information we separate the extract into some fraction. Yeah, and then each fraction has different activity. Like here, we select the fraction 9 as the most active one. This one. Yeah, the fraction 9, the active fraction we make in OPLSDA as one group, yeah, and then in active fraction in this area. Yeah, from fraction 9, we try to check the compound and we found this is gallic acid, protocatechin acid, and then EGCG, myrcetin rhamnoside, glutamic acid, and also the unknown compound. Yeah, this is the compound in fraction 9. From there, we try to make sure that this is really the alpha glucose inhibitor using in silico analysis. And we can find the binding energy, even not as good as acarbose, but almost all the compound is active, especially until number 6, until number 4, I mean. This is the data, the last data for diabetics, and also we already make like monograph of biopharmaca for diabetic mellitus, especially research in Indonesia, not Indonesia, in IPB University. Yeah, we make like summary like this. That most of our native Indonesian plant here has different way to be an anti-diabetics. Okay, so next I will move to body weight and inhalation of native Indonesian herbal medicine. So this is actually my dream. I want to be slim without doing exercise. Maybe this is not good, but this is my dream. The result start because plant, especially plant that has essential oil, they can be used for aromatherapy to enhance the body health and also beauty. Yeah. These effects are sensory examination lacking scientific evidence. Yeah, only few information about its active component. So since every other researcher only search for their own medicinal plant, so I want to focus with our medicinal plant. But the background is based on the information of lavender and also grapefruit oil for anti-obesity. Yeah. How grapefruit oil become anti-obesity? Because this essential oil will goes to the olfactory receptor and comes to our hypothalamus and will make signal to the sympathetic nerve to us release of noradrenaline. And after that, lipolysis will be decrease. Yeah. So when lipolysis decrease, it means our body become sorry. Okay, sorry, move ya. This is grapefruit. Yeah, first is grapefruit. For grapefruit, adrenaline is released, and then lipolysis is increased. When lipolysis increase, especially happen in brown adipose tissue, triglyceride, yeah, become free fatty acid, and UCP1 will give a heat, so our body become warm, yeah, become warm. At that time, the triglyceride in our body will change become free fatty acid, so we will be decrease our body weight, and sometimes decrease the appetite. But it will be happen when we smell or inhale the grapefruits. Different result when we inhale lavender. Yeah, when we inhale lavender, it become decrease the lipolysis. Yeah, so not release the heat, but increase, not increase. So our body become more relaxed. Yeah, become more relaxed, and so body weight will be increase and appetite also increase. So I will check for our Indonesian medicinal plants that have essential oil. One is citronella. Yeah, this is citronella oil. We always use it for analgesic, anti-mosquito, candle, and also soap aroma. It consists minimum 2 compound. Yeah, one is beta R citronellol, and the other one is beta citronellol. Based on this data, so I give the mouse, yeah, mouse with, not mouse, this is we use a rat, yeah, rat that consume high fat diet, and the other group, they consume high fat diet, but they inhale citronella oil. The other group, they inhale R-citronellol, and the last group inhale citronellol. Interestingly, the data, this is feed consumption, actually not so significant different, but for fecal and urine excretion after treatment, yeah, in a group of citronella oil, they increase the fecal and urine excretion, yeah, but not significantly different. And citronella decrease, yeah. This is not the same result. The fecal is decreased. It related also with the this is different, yeah, significant different. The consumer feed consumption is decreased on the group of citronella. So after this, we check for the cholesterol level, yeah, cholesterol level of each group. Sorry, yeah. Here the result, and the lowest cholesterol level we can find in citronella group, but the lowest triglyceride is in 3 other group, and the other 2 for hepar activity is not significantly different. And from here, we check the activity for sympathetic nerve activity. Yeah, this is we try to open in this area for the mouse or rat. Sorry, this is rat, and we check the connection in the nerve connection to brown adipose tissue and to scapula. And we put the nerve here in the silver wire, yeah, silver electrode, and we check the data using oscillator. So this is the information when when we put the nerve in the electrode, we can see the signal in bioelectric amplifier and goes to oscilloscope. This mean there is signal, and then next we calculate all the data in Power Lab and put in our PC. So the data become like this. This is one signal to be one line like this one. This data we call as spike histogram. Yeah, histogram. So this is the when the electrode no nerve, so no signal. This one is we call as base peak. But when we put the nerve, we can see the spike of nerve activity. Yeah, we can check the nerve in activity when we put the nerve in the electrode. The result shows here when we give this is the rat only inhale water, yeah, but this one after inhale citronella. So the activity decrease and the temperature of the brown adipose tissue also decrease. And different with citronella, it's a little bit increase, and also this one also a little bit increase. And this one for oil, the mixture, little bit decrease, but not significantly different for the sympathetic nerve activity. Okay, this is the brown adipose temperature we check inhaling because of inhale the citronella oil. I already informed here already, yeah. So the summary is here, no liver function was damaged by inhalation of citronella oil, R-citronellol, and also beta-citronella, R-citronellol and beta-citronellol. And then inhalation of citronella oil, specifically beta-citronellol, decrease weight gain by increasing the sympathetic nerve activity, which was related to the decreased appetite. Yeah. And the cholesterol level also decrease in the rat inhale beta-citronellol. This is the way that the inhalation can decrease the body weight. Yeah. And the other we check mostly is gymborase essential oil. Yeah. From this one is lengkuas atau lengkuas merah red. Oh, what is it? I forget in English. Yeah. And then temulawak, kencur, temu kunci, bangle, jahe, and also lempuyang gajah. Interesting data. Some oil can decrease the body weight, especially ginger, until 77.1% compared to the positive control. But lengkuas only 2.8%, compared to positive control. The other oil increase the body weight, especially lempuyang gajah. They can increase the body weight become 17.2%, Oh, so this all the information from me for the closing. Indonesia still have many natural resources to prevent the NCD, so more research and product could be developed. I think collaboration will make it happen soon. Thank you very much for your kind attention. Okay, I send back to Pak Made.
Right. Right. Thank you, Professor, for your brief explanations. Maybe we can now invite if there are any participants who wants to ask questions. Any questions? Right. Probably I will kick this off to ask question to Professor Imanida. From your explanations, I saw there are many native herbals. Uh, is it already mapped by the TROP-PRC Indonesia IPB that almost all herbals being mapped or being analyzed for its chemical components, especially for the diabetic, as you said previously, Professor?
Okay, thank you, Pak Ahmad Yani. Yeah, so not all compound we already collect the information about. Not all compound we already collect the information. We only collect the information for specific that we think are interesting. If no, still many things we have to do, Pak Ahmad Yani.
All right. So for the last slides that you are describe about the oil inhalations, is it also is it for only for the body weight decrease or any other, let's say, diseases probably, Professor, that can be affected by this oil?
Yeah, I think this oil in.
Or is there any possibilities for these oils to also reduce NCD's prevalences probably?
Yeah, still we have to do the research, Pak Ahmad Yani, because now we are thinking first for this oil is mostly for pulmonary disease or COPD is the best maybe, because when we inhale it will be goes to our olfactory system and then goes to the lung, for example. So we think this is related also for lung cancer, but no, not we did not do the research yet until now. And also for the pandemic, mostly everybody asks you to inhale like minyak kayu putih ya, cajuput oil. Maybe this one can make the bacteria in our respiratory tract will be dead, maybe some of the bacteria or viruses. So this is also the way that we can do the research. But bacteria maybe not for NCD ya, the other for penyakit menular justru ya. Is it okay Pak Ahmad Yani?
Thank you for the answer. Is there any questions from the audiences probably?
Yeah, Professor. There's a question from the audience.
Okay.
Thank you for the time.
Yeah.
Professor, I just want to ask about which Indonesian herbal is effectively to decrease for blood pressure in hypertension people. In
Okay.
some, there are so many medicine herbal Indonesian herbal that can use to combine to control the blood pressure. But from your presentation, maybe I can see it. I cannot see it. It's all about
Diabetic.
the NCD, only for NCD. And is there any recommendation for our PUI that we can more develop the herbal Indonesian to increase our health. Thank you.
Okay, thank you. Dengan Ibu, sorry, I cannot catch the name ya.
All right, thank you for the questions. That's from Miss Annie.
Miss Ani, okay.
Professor. So the question is, from your presentation, that's only diabetic. Probably it's your focus research, but one of the questions is that our PUI topics is hypertension. So do you have any recommendations which herbals that can be developed probably to increase our health? That's please, Professor.
Okay, thank you very much, Bu Ani. Ya, we did also the research related to hypertension, but since I'm not majoring in this theme, so I did not present this one. But the one that I know now, Prof. Dia Iswantini try to register the product for hypertension. I forget the insight is what plan ya. I worry I mistake, so I cannot mention in detail. The one that I remember is from Mahkota Dewa ya, Mahkota Dewa Pareria macrocarpa, the leaf but not the seed and not the fruits ya, the leaf. The one that we did the research, Ibu Irma Suparto, if I'm not mistaken. But for Prof. Dia product, I forgot the plant name. I'm sorry, I will inform you later. I think still many Indonesian plant that we can use for hypertension to prevent the hypertension. But since I'm not the one that do the research, so I cannot talk in detail for that one. So sorry. That's Pak Ahmad Yani.
Thank you, Professor, for the brief answer. Is there any questions from the audience? Yeah, the audience may raise hands if there's any questions for the speaker. No? All right, probably there is no other questions, Professor, from the audiences. So maybe let's wrap up from the second session, probably that Indonesia, as described by our professor, has a lot of herbals that can be developed in order to probably to contribute to our health. And there are native herbals from Indonesia that can be developed in order to assay its chemical compounds that can contribute to our health. Is there any additional things you want to add up, Professor, to close this session?
Okay, I think for closing this session, we still have many chance to utilize our natural natural resources, yeah, especially also for hypertension. If I'm not mistaken, celery, yeah, the one that used by Prof. Dia to make a product for hypertension. So maybe let's do it together. We can make it happen soon. I think like that.
Yeah,
Thank you
sure.
very much for everybody.
Yeah, thank you
Assalamualaikum
very
warahmatullahi
much for your
wabarakatuh.
presentations, Professor. Give
Waalaikumsalam.
applause to our Professor
Thank
Imanida
you for
Batubara from
inviting.
IPB University. We highly appreciate your attendance here, joining us today in the second session. And we really thank you for your speech. It's really useful and knowledgeable for us. And maybe we see you later on next occasion, probably. Thank you very much, Professor Imanida Batubara.
Thank
And
you very
I stand it back
much.
to moderator. Professor, we take picture together, probably. Yeah.
Okay.
All right. Yeah, please, audience, you may on your camera if it's possible. All right, 3, 2, 1. Okay, page 1, okay, and second page. 3, 2, 1. All right, it's enough. Thank you, Professor Imandira Batubara, for your
Thank you everybody.
attendance.
Mohon maaf lahir
I
batin.
hand it back to our MC, Miss Siti. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you very much, Professor Irmanida Batubara, for the presentation. Ladies and gentlemen, now we are moving to the final speaker of the webinar. The next will be Professor George Statue, who will deliver the topic of methods of measuring blood pressure and applications to diabetes. His third session will be moderated by Mr. Ahmadiani again. To moderator and speaker, please take your time.
All right, ladies and gentlemen, still joining with us, thank you very much. The third session will be delivered by our professor from Greece, from the University of Athens, but at the moment, he still try to connect with us to the Zoom meeting, and we're still waiting for him to connect with us. But previously, I will I will show to the audience his CV. Yeah, this is the CV of Professor George Stergio, MD, FRCP. He's a professor of medicine and hypertension. He's the chairman at the Hypertension Center Stride 7 School of Medicine at the University of Athens. He's a secretary at International Society of Hypertension. He also at the World Hypertension League as a special envoy for blood pressure management. He's also International Pediatric Hypertension Association Executive Committee member, and also he's joining International Organization for Standardization, especially as a committee member on blood pressure monitoring equipment, and also he's past president at Hellenic Society of Hypertension. He's also secretary at International Society of Hypertension, along with a long role of him. And the last, probably, he's elected as Fellow Royal College of Physicians, or FRCP, from the Glasgow, UK, and also he's an editorial board member on 11 PubMed journals, including reviewers on 58 PubMed journals, and also McMaster University's Health Information Research, PRMed Journal, Publishing Group, etc. And he has H index more than 79 with citations about 52,000, including citation in Hypertension Society's guidelines. So we're still waiting for him to joining us today. Yeah, I still cannot say his name at the participant list, so probably probably I will send to our MC again. Yeah, maybe for take a break, probably. Yeah.
Ladies and gentlemen, we apologize for the delay due to technical problems for the speaker from Greece. And due to technical problems from Prof. Sergio, And we will take a break for 30 minutes. Thank you.
Kalian nggak sesuai.
Yeah.
Ay, Paul, nakamari nga na? Nakamari nga na. Yan nakamari nga na. Paul, yan nakamari nga na. Nakamari? Ano yan nakamari?
Good morning, Professor.
Good morning, Professor Sergio. Good morning, Professor Sergio. Are you able to hear?
I cannot hear you.
All right, thank you. We can hear your voice as well. All right, we already introduced your CV to the audiences, and now if it's possible for you to start the talk, yeah, it will be convenient if you already begin. Thank you. Professor, please, time is yours. Thank you.
Thank you very much. Can you hear me?
Yes,
Yes.
it's clear.
Thank you. And can you see my slides and full projection?
Yes, it's already can be seen.
Okay, thank you. Thank you very much for this invitation to contribute to your meeting and talk about blood pressure measurement in the office and out of the office using home or ambulatory monitoring. My name is George Sergiu. I'm a professor of medicine in the University of Athens in Greece. So the history of measurement for blood pressure is an old story, and this priest, often held since 1677, was the first to make this measurement by putting a glass tube in the artery of a horse. With his experiment, Stephen Wales first measured the pressure in the blood, in the circulation, and also observed its dynamic variability. So this measurement has been very important for us because we were able to have clinical hypertension. It is this measurement that allowed us to open the chapter of clinical hypertension. It is the only method and the basis for diagnosing and managing hypertension. It is routinely used to initiate or to rule out costly investigations and long-term drug therapy. And inadequate methodology or use of an inaccurate blood pressure monitor can lead to overdiagnosis, which means unnecessary treatment for a long time, or underdiagnosis, which actually is related to preventable cardiovascular disease. So blood pressure measurement has always been vital for hypertension, but today it is more important than it used to be before. And this is because today we ask our physicians to have lower blood pressure targets, and the measurement accuracy is crucial. This is to achieve optimal control and prevent adverse effects of overtreatment. And my presentation, which will be largely based on this paper, these are the 2021 European practice guidelines for office and out of office measurements. You can see this has been a paper prepared by not only European experts, but also from Australia, United States, China, and several other countries. So it's a universal, purely international paper, which is not saying why we should do what we should do, but it's a practical paper, just what we need to do for proper measurement. So currently we have the classic office measurements widely available, but these measurements are taken in artificial setting, which can affect the blood pressure of the individual. And we can only take 1, 2, or maximum 3 readings for 1 or 2 visits. Home blood pressure is different because first, this is taken to the usual environment of each individual at home. And also it can provide us many measurements for days, weeks, or months. And we have also 24 ambulatory monitoring, which is another different method because measurements are taken on a usual work day, not in the office, not only at home, but at work, during routine daily activities, at home, and during sleep. And this method can give us almost 100 of readings within 24 hours. So we have 3 methods, and these are complementary. Office is everywhere. I don't know about your country, but in my country, in Greece, all patients will bring us their home measurements even before we ask for them. Ambulatory monitoring is a problem because it's not accessible to many healthcare settings. So this is what I'm going to talk today. Office, home, and ambulatory measurements, starting by the office measurement. And this is supposed to be the cornerstone for diagnosis and management in hypertension. It is most widely used. It is available in almost all medical settings. In many settings, it is the only method available. Many settings do not have access, and it's not only about countries, but even in the United States, even in the richest countries, there are some settings where decisions will be taken based only on office measurements. And it is the most well studied. The strongest evidence we have is about this method, and this is the method we used for the classification of blood pressure, the thresholds of treatment, and the goals. It's not home, it's not ambulatory, it is office. But this method has serious limitations. The most important one is us, the healthcare professional, the human observer, error, and bias. Second, it is poorly reproducible. Office blood pressure can change from visit to visit without any change in treatment. It is reduced with placebo, and it is affected by the so-called white coat and masked hypertension effect. So let's say that we have our patients and we take proper office measurements and out of office, ambulatory of home. So if you assess your patients in the office and out of the office, you find a nice correlation within this measurement, a high correlation coefficient. But if you go to apply it to the diagnostic thresholds, you realize that there are some people who have high blood pressure everywhere. They are hypertensives. There are those who have low blood pressure everywhere. These are normotensives. But there are people who have high blood pressure only in the office, the so-called white coat hypertension phenomenon. And we have another more tricky phenomenon, which is called masked hypertension. These are patients who will come in our office. They have low blood pressure in the office, but out of office, it is elevated, masked hypertension. And even if we are very careful in taking both the office and out of office measurements, there is diagnostic disagreement between these methods in 30% of our patients, either untreated or treated. And I have to make a statement here. If properly taken, these methods, in case of disagreement, the correct information is usually provided by out of office, not office measurements. And you can see here in our center, using either ambulatory or home measurements, among these patients who are referred to our clinic for suspected hypertension, more than half have high blood pressure in the office and out of the office. 15 to 20% have low blood pressure in both measurements, but we have 16% white coat hypertension by both ambulatory and home measurements, and we have 10 to 15% masked hypertension. So even with careful measurements in research settings, almost 30% of patients are going to have white coat and mass hypertension, and you need out-of-office measurements to identify these cases. Is this important to identify these patients? Well, this is a meta-analysis of several studies, the IDHOCO by Professor Jan Stuyssen in Belgium. He included from our studies more than 6,000 people, and these were followed for 8 years. And we analyzed separately untreated and treated hypertensives. And you can see here that the risk of white coat hypertension is a little higher than normotensives. There's no difference in treated, but what about masked? Masked hypertension has similar risk to untreated, to uncontrolled hypertension, and particularly in treated individuals, masked hypertension appear to have even higher risk than uncontrolled hypertension, untreated hypertension. Why? Because the doctors do not see the elevated blood pressure in the office and leave it untreated. And this is a case with masked hypertension. This is a banker. He's a friend of mine and my patient. And you can see this is his blood pressure at work. 170 on 100, so quite high blood pressure during 6 or 7 hours at work. And then he comes to my visit. It's always the last evening visit, and his blood pressure is low in my office. And at home, his blood pressure is low, and during sleep. So this patient has high blood pressure only at work. If you don't do ambulatory monitoring, you will never find this uncontrolled case. And this is a poster we have in the European guidelines for blood pressure measurement. And you have seen several of these for many years, showing how to take a proper measurement. And there are so many sources of error to a proper measurement related to the environment, to talking, temperature, time of rest, the position of the arm, the legs, the cuff size, the device. So there are so many sources that can lead to improper measurement, and all of them should be taken into account. This is why Professor Pickering in the United States several years ago has questioned the usefulness of doctors taking blood pressure in the clinic, mainly because of masked hypertension. So we realized in the recent years that we have different types of office measurements. When we say we measure blood pressure in the office, we have to say how we measure blood pressure in the office. For example, we have the usual casual clinical practice measurements with your auscultatory method. They seriously overestimate the blood pressure of the patients. We have clinical practice automated attended measurements, which are lower. They still overestimate blood pressure, but they are lower than auscultatory. We have clinical practice automated measurements, which are unattended, which means you leave the patient alone in the room and the device takes 3 or 4 or 5 measurements. These are lower. Here you underestimate office blood pressure. And we have the research settings with any method, research setting measurements, where in very careful readings underestimate blood pressure. So we have different methodologies for taking office measurement. And these methodologies use different thresholds and lead to different conclusions. So when somebody tells you blood pressure was in the office was like that, you have to ask, well, how did you measure blood pressure in the office? And for office devices, there are many devices. You can see these devices are electronic devices designed for the office, and they're quite expensive. Most of them have cost something like 500 euros or longer. This is an electronic devices. This is not a mercury device. These are not mercury devices. These are electronic devices, but these are manual auscultatory devices. And I have to make a statement here. These expensive devices are not more accurate than our home measurements. So if you don't want to pay that much for your office device, take a validated home device for office measurements, no problem, same accuracy. So the European guidelines recommend that in the office, we should prefer electronic devices. So today, we don't favor auscultatory devices as our first option for the office, but electronic ones. We recommend upper arm cuff devices, not wrist devices, and we want these devices to be properly validated using an established protocol. So moving to ambulatory monitoring, this is an 24 ambulatory monitoring using an intra-arterial method. So it is bit-to-bit measurement, each 100,000 readings for 1 day. And you can see the dynamic variation of systolic and diastolic blood pressure. You can see the prompt drop during sleep and the rise in the morning. So this is what we want to know. This is what we're interested in because this is the burden that the cardiovascular system sees from high blood pressure. If you take blood pressure in the office, you take this very limited picture. If you take at home, you have 2 windows, but again, very limited information about the 24-hour blood pressure profile of the individual. So here you have a picture and you have small snapshots of the picture. This is what you do with office home measurements. Very small, very little information, but with ambulatory monitoring, you get the entire picture. So ambulatory monitoring is the best method for hypertension diagnosis. It provides multiple blood pressure readings in routine daily activities at work, at home, and during sleep. And it's the only method that tells us about nighttime blood pressure, which is important. It identifies the white coat hypertension and mass hypertension phenomena. It evaluates 24-hour blood pressure control. It is considered today as the best method we have for diagnosing hypertension. But there are limitations. It's not widely available. Many doctors will tell you, I don't have access to this method. It's expensive. I don't know why these devices are so expensive. It is inconvenient for some patients to wear at work and during sleep. It is not perfectly reproducible, although more reproducible than office measurements. So how to use it? First, use it only on a usual work day. We never recommend ABPM when a patient is admitted in the hospital. Take readings every 15 to 30 minutes, not only during the day, but also during the night. When you finish, you need to have at least 20 valid awake and 7 valid asleep readings. Otherwise, the ambulatory monitoring should be repeated. And the first criterion is the average of all readings. It should be lower than 130/80. This is the primary criterion, and then you can go to day and nighttime dip. I'd like to talk a little bit now about diabetes. This is a large database from Spanish where they had 15,000 treated hypertensives. They were taking treatment, and blood pressure in the office was controlled. And then they used ABPM to show that 31 of these who appeared to be controlled in the clinic had masked uncontrolled hypertension. And who were these patients? Most commonly men, high risk, and diabetics. So diabetics have a much higher risk for having mass hypertension than non-diabetics. Solely daytime elevation was found in 13%, and nighttime elevation, isolated nocturnal hypertension, was 24%. So ABPM should become routine to confirm blood pressure control, particularly in high-risk groups, namely diabetics. And this is another study from the IDACO database, again by Professor Stuyssen in Belgium. And you can see the prevalence of mass hypertension in non-diabetics was 19%, but in diabetics was almost 30%. And not only the prevalence was higher, but you can see untreated mass hypertension had double the risk of normotensis. So it's important to diagnose these people among diabetics with ambulatory monitoring. In this study, it's a small study, but here the prevalence of mass hypertension diabetes was almost 50%. And these patients had increased left ventricular mass, which was higher than normotensis, but lower than sustained hypertensives. And left ventricular diastolic dysfunction was similar in mass and sustained hypertensives as less so than normotensis. So mass hypertension in diabetes is associated with cardiac organ damage. So this is another study from a Spanish registry, including 12,000 people, and you see the abnormalities of ambulatory blood pressure in diabetes. First, diabetics have higher systolic daytime and nighttime the blood pressure, despite the fact that they were using more antihypertensive drugs. Second, more frequently they had uncontrolled blood pressure, particularly during the night, which means you need ambulatory, not home measurements to identify these cases. They were more frequently non-dippers than non-diabetics, and more frequently had mass hypertension. You can see the vital importance for out-of-office blood pressure evaluation in these people, particularly during the night. And this is another study showing that the loss of the blood pressure decline was associated with increased mortality rate in both diabetes 1 and 2. And when you had combined non-dipping and renal impairment, you had the highest mortality rates. So these data show that there is a major role for 24 ambulatory monitoring for diabetics for selecting those patients who are at greater risk. And this is another study in elderly diabetics who were, again, the blood pressure non-dipping was associated with high risk of cardiovascular events and stroke. And when you had combined blood pressure and pulse rate non-dipping, you had the highest risk of cardiovascular events and stroke. And this is another small study in diabetes 1. And these are young people, adolescents, where the damage is only starting. And you can see here increased systolic nocturnal blood pressure, which can identified by blood ambulatory monitoring, precedes the development of microalbuminuria. And the normal nocturnal blood pressure decline was less likely for microalbuminuria development. So you can see the vital role of nighttime blood pressure in diabetes. So the European Society of Hypertension, in its position paper for ambulatory monitoring published several years ago, they stated that ambulatory monitoring is mandatory in diabetes because mass hypertension and non-dipping are very common in these people. So diabetes has a special 24-hour blood pressure profile characterized with mass hypertension, nocturnal hypertension, and non-dipping, and these phenomena can only be identified by 24-hour ambulatory monitoring. Now moving to the home blood pressure measurement. The European Society of Hypertension recommends this method for the initial diagnosis and for following treated hypertensives. For initial diagnosis, home blood pressure can be used to confirm the diagnosis, and it's important to detect the white coat and mass hypertension phenomena. But the most important role is treated hypertensives. The European guidelines say use home blood pressure measurement in all treated hypertensives, unless they can't do it. It can identify the white coat and mass hypertension among treated patients, It's useful for treatment titration, to monitor long-term control, and those patients who self-monitor their blood pressure at home, they have better long-term compliance with treatment. And we have several studies to show that. You can see here meta-analysis, a systematic review, 24 randomized controlled trials, 75 people. We know that those people who have a home monitor and self-monitor their blood pressure have a small but significant overall effect in improving their medication adherence and improving their blood pressure control. This is why Tom Pickering several years ago called for action to use and reimburse the home blood pressure monitors as much as we reimburse the glucose monitors for diabetics. We should reimburse home monitors for hypertensives, because home monitoring has the potential to improve the quality of care while reducing the cost of care. And you can see here what patients believe. It is important how patients think about what we do. This is one study on the left in UK, and on the right one study in Greece from our center. You can see in UK 80% of patients prefer home measurements. 19% prefer laboratory monitoring. And you can see here, whatever you ask the patients, is it comfortable, it is less uncomfortable, they find home blood pressure more comfortable. In the study in Greece, how easy it was to perform home blood pressure versus ambulatory, both patients preferred, they found it more easy to do home measurements. What about devices? For home monitoring, we should use electronic upper arm cuff devices, which are properly validated. We should prefer devices with automated storage and averaging, or mobile phone or PC or internet connectivity, because we want to have an overview of all readings in an objective way. Wrist devices are not recommended unless arm cuff measurement is not possible. Auscultatory devices are not recommended for use by patients at home. Cuffless wearable devices not recommended for hypertension management at the present time. How to measure blood pressure at home? How many measurements? It's incredible how many research and published papers we have on this research question. The optimal schedule is to ask our patients to measure for 7 days. On each day, they take 2 measurements in the morning and 2 in the evening. After 5 minutes rest, 1 minute apart. The minimum schedule is 3 days with 12 readings. Less than that, you need more measurements. So optimal 7 days, minimum 3 days. And how do you interpret these readings? The first day when the patient starts the measurement, it's always higher and unstable and should be discarded. So we calculate the average of all readings after discarding the first day. We don't look at individual readings if they are very high or low. They have very little diagnostic accuracy. And the average of all readings should be lower than 135/85. So you see, the threshold for diagnosing hypertension at home is lower than at the office. And this is a form we give to our patients, and we tell them when the next time to come for the next appointment. A few days ago, fill in this form with your home readings, so as we have a picture of his blood pressure at home. And we give recommendations how to measure before each visit, and 7 days and so on, which I already told you, and how to measure in the long term between office readings. In this case, we tell them measure once or twice per week or month. But home blood pressure has a weak point. And ladies and gentlemen, we cannot rely on these kind of papers in order to make decisions for long-term treatment. We cannot say this patient is not hypertensive because these papers say low blood pressure when we have high blood pressure. We need objective measurements. We know that patients select which readings they are going to show us. That whenever they monitor something at home, they are selective. They do not cheat. They just think some readings are not representative. They tend to delete the high readings. So to eliminate this error, we need electronic devices with automated memory or PC link to have an objective reading, or home blood pressure telemonitoring will become cheaper, will be the 21st century solution for evaluating hypertension. And there are several accurate devices in the market by several major manufacturers, and they can give the average of all readings automatically and the graphics of the blood pressure for weeks or months, and you can have them in the mobile of your patients, the total average, and these will become affordable for more of our patients quite soon. And they can send these readings to our files, and so this is an objective way to have continuous monitoring of blood pressure of our patients. And you can see here a meta-analysis of 23 randomized controlled trials for home blood pressure telemonitoring, which shows that this method can be a useful tool to improve the hypertension control, but still is costly, and we hope this will change. So I said that 24 ambulatory monitoring is the optimal method for assessing the diurnal blood pressure variation. So we have been saying that ambulatory monitoring is the best method for looking at nighttime blood pressure. Not anymore. Today we have low-cost home monitors which can take automated measurements during nighttime sleep. You can see a study we published where a low-cost home monitor was compared with ambulatory monitoring. You can see similar day and nighttime readings and similar proportion for detecting non-dimmers with ambulatory monitoring versus home for nocturnal blood pressure. I'd like to talk a little about devices. This is an amazing device, the mercury sphygmomanometer, which has stayed with us for almost a century. In this century, we have invented the automobile, the plane, we have put men on the moon, we have invented the microchip, and we still have this device in our office. This has been an amazing device. More important, this device is still being used as a reference method for testing any novel technology for blood pressure measurement. But today is the era of electronic monitors. The European Society of Hypertension recommends electronic devices not only for home and ambulatory, but also for office use. The problem is that more than 10,000 electronic devices on the market, and less than 10% are properly validated. And another problem is that some devices which are accurate in adults, electronic devices, may not be accurate in special populations, particularly children and pregnancy, and separate validation is necessary. So the STRIDE BP is an international organization which is endorsed by the European Society of Hypertension, the International Society of Hypertension, and the World Hypertension League. And these organizations provide lists of which devices are accurate for healthcare professionals. And you can see that less than 10% of the electronic devices on the market are accurate. And the STRIDE BP, you can download lists of accurate devices for home use, for office use, for ambulatory use, and separate the list of accurate devices specifically for children and for pregnancy. And this website has reached more than 200,000 views from 190 countries. I'll talk briefly about cuffless continuous measurement. This is a great promise that you have a device on your arm, on your wrist, and it measures blood pressure for days or weeks. It may provide a complete picture of the 24-hour blood pressure profile for long periods of time, weeks or months. and it may accurately reflect the burden of blood pressure on the heart and the vascular. And you can see several devices. This is the Actia device. This is the Samtuk device, the Measures device, several devices claiming that they can accurately measure blood pressure without a cuff. And there are also applications, very attractive applications on your mobile phone. You put on your finger, you have the blood pressure reading. It is important to realize that these devices are different. They use different technologies and they have different intended uses. In August this year, the European Society of Hypertension published a statement to inform healthcare professionals about where we think we are today with all these attractive cuffless blood pressure technologies. The conclusion is that there is a large number of such devices on the market, and some of them have clearance from some organizations. They have great potential, but at the present time, their accuracy and usefulness is uncertain, and they should not be used for medical decisions, diagnostic, or treatment. So, what do the guidelines say for all these readings? You can see the European guidelines. They say if you have in the office high normal blood pressure, you have to consider mass hypertension with out-of-office measurements. But even if you have high blood pressure, you need to confirm with out-of-office readings to exclude white coat hypertension. So in most of the cases, particularly in those people whose office blood pressure is close to the diagnostic threshold, you don't have the accuracy of measurement in the office to make a diagnosis, and you need confirmation with out-of-office measurement, ambulatory or home. And you can see the American guidelines. They say that ambulatory and home readings are recommended to confirm the diagnosis. So the diagnosis is not made in the office. And not only for the diagnosis, whenever you think you should titrate treatment, you need to confirm with out-of-office measurements. And this applies to both untreated and treated individuals. Why don't we use them? You can see here data from the United States. For home blood pressure, there are concerns that patients may not measure correctly. There are concerns that some people may become preoccupied with blood pressure and measure many times every day, and sometimes may make change treatment on the accuracy or on the basis of these measurements. And there is a problem with device affordability in some populations. For ambulatory monitoring, the issue is the cost. It's an expensive method. It requires a lot of time by the physician to initialize the device, to explain to the patients, to download the device, a lot of time. There is a lack of infrastructure in including access to these services, and there is lower reimbursement. But ladies and gentlemen, another very important thing from the United States is that they say that healthcare providers may lack confidence in their skills and knowledge to interpret ambulatory and home measurements. And this is our responsibility to teach primary care physicians, and this is why our meeting today is so important. So I'm close to the end. You can find this paper. It's a practical paper with tables and forms for clinical use for all the methods. You can see here one page, how to do office. You can put it in your clinic. One page, how to use ambulatory monitoring. One page, how to use home measurements. All practical recommendations for practicing physicians. And these guidelines have been translated in many languages to be widely used for clinical practice. And for those of you who wish to have a proper training on how to use these methods, StrideBP has a free of charge training modules for office, home, and ambulatory monitoring. It is 20 to 30 minutes for each module. And when you do all three of them, you can take a test, you can take a certificate for home, office, and ambulatory monitoring. Thank you very much for your attention.
All right, what a brief presentation, Professor. So, yeah, here several questions already received by me as a moderator. Maybe, yeah, from the audience. Yeah, there's a question from our audience, Professor. Maybe I will invite her to speak and ask directly the questions to you. Miss
Yes,
Ani, please.
Thank you for the time and thank you for your explanation. Very comprehensive. And I want to ask about in your presentation, so that if we want to measure the blood pressure, it's better in the morning and the night and at the night. In my, as I know that circadian rhythm of the body in the night, naturally our blood pressure is increasing. So the first I asked you, why did we measure the blood pressure at the night?
Yeah, thank you for this question.
The second,
do you want me to respond right now, or you have a further question?
yes, the second question is how to decrease our limitation in our research to measure the blood pressure when we have a lot of many samples. So how we can control, because there are so many confounding factors that make blur result, because every person is unique. So how to limit, to decrease the limitation that our result is valid and measurable. Maybe that's from me. Thank you.
Okay, thank you. I think both these questions are very important. First, because there is a lot of evidence in the recent years that nighttime blood pressure is a very important aspect and probably the most important blood pressure measurement in terms of predicting cardiovascular disease. So it's important to measure that. In the question which blood pressure is more important, in research trials, nighttime blood pressure is the most important. But for clinical practice, we need a lot of information. I would say you need several readings in several times of the day. And the most important thing is that you need many readings. If you do office, you need 3 visits. If you do home, you need 7 days. If you do ambulatory, you need 2 24-hour recordings. So nighttime is the most important, but it's not enough for the individual. For the individual, we have moved from measurements to profiles. So you need several readings to have a good picture of the individual. And this goes closely to your second question, how can we have a good evaluation of our individual patients? And in this case, I would say to be confident for the blood pressure of the individual, you need 2 methods that agree. I discussed 3 methods, office, home, ambulatory. In my practice, if I have office and home, or office and ambulatory, or home and ambulatory, and both of them agree, then I'm confident I know where blood pressure is an individual. If I have two methods and they disagree, then I go for the 10th method. But I do not trust any of the methods alone. So for me, you need two methods agreeing, and then you can be sure about the blood pressure of your patient.
Ada nanya dia.
All right, another question probably from Mr. Budi. Mr. Budi.
Dia tolong diterjemahin deh.
Boleh, you may say
Nanya.
in bahasa and we will try to
Mau nanya
translate in English.
kalau seseorang sudah terdiagnosis hipertensi, apa hidupnya harus selalu ketergantungan obat-obat hipertensi deh? Terus
Alright.
bagaimana dengan obat-obatan yang diketemukan dengan herbal, apa bisa menyembuhkan hipertensi deh? Terima kasih deh.
All right, these are interesting questions, Professor. So the first question from Mr. Budi is, he asked if somebody has been diagnosed with hypertension, is it going to be a lifelong disease in his or her life? So that's the first question. If somebody has been diagnosed with hypertension, is it a lifelong condition in his life or her life? And the second one, if that person consume like herbal medicine or traditional medicine, is it good for him or her in order to cure or probably to stay to maintain his normal conditions? Yeah, that's the questions from Mr. Budi. Please kindly answer, Professor.
Thank you for these interesting questions and useful to discuss. In regard to referring to my previous response to the other questions, it's not easy to make diagnosis of hypertension. You need measurements, several measurements, and two readings. So, about having a permanent diagnosis for life, there are two scenarios. One is your diagnosis accurate? Because in many cases, the diagnoses have been carelessly made on the basis of few office readings, and then after a few months, you realize that there is no high blood pressure. But this is not that hypertension disappeared, but that the diagnosis was wrong in the first place. But if your diagnosis is accurate, using office and out of office measurements, probably this diagnosis is permanent, with one exception. If there was something that happened to the patient which reduced his blood pressure, for example, lost a lot of weight, a seasonal change, it was winter and now it's summer where blood pressure is lower. He used a drug which increased blood pressure and stopped that. So the diagnosis, if properly made, is permanent for life, one. And second, unless something has been done which effectively changed blood pressure, the diagnosis will remain permanently. Now for herbal, I'm glad you made this answer. I am diagnosed with hypertension. What I'm going to do? What is the goal of treatment? The naive answer is that the goal of treatment is to reduce blood pressure. No, this is not the goal of treatment. The goal of treatment is to prevent the risk associated with blood pressure. So if you come to me and tell me, I have this stuff, drink this every morning and blood pressures go down, I will not take it. I don't want something that will reduce my blood pressure. I want something which have outcome data with 1,000 patients, which says that if you drink that every morning, your blood pressure will go down and cardiovascular disease will prevent it. If you don't have data for a drug or a herbal or everything that shows not simply that it reduces blood pressure, but that it will prevent cardiovascular events in the future, I will not take it. So the drugs we use today are not those who reduce blood pressure, are those who effectively prevent cardiovascular events in the future through the reduction of blood pressure. If you don't have this data, don't take anything every day.
All right, that's brief explanation. Please allow me to translate it first, Professor, to Mr. Budi and other audiences. Jadi mungkin Bapak Ibu, yang pertama dari pertanyaan Pak Budi adalah apakah jika seseorang sudah didiagnosis hipertensi, apakah itu akan menjadi kondisi yang berlangsung seterusnya begitu? Nah, ini sebetulnya jawaban dari beliau ini bergantung ke dua skenario. Yang pertama, apakah diagnosis tersebut sudah valid atau tepat? Apakah misalnya pengukurannya tadi seperti yang beliau jelaskan sudah dilakukan misalnya untuk office itu 3 kali visit minimal, kemudian untuk ambulatori misalnya 2 sampai 24 jam begitu, kemudian yang untuk home measurements 7 hari berturut-turut misalnya. Nah, ini harus diketahui dulu apakah memang diagnosisnya ini valid atau tidak. Kemudian yang kedua skenario-nya ini jika memang hasil diagnosis tersebut akurat, maka kondisinya ini akan permanen. Kecuali dengan satu exception tadi, kita melakukan upaya-upaya tertentu seperti tadi misalnya menurunkan berat badan, kemudian melakukan aktivitas fisik dan lain sebagainya. Jadi itu yang beliau sampaikan terkait pertanyaan pertama. Kemudian untuk pertanyaan kedua, beliau menjawab bahwa beliau menyarankan jangan ambil advice tertentu, misalnya dengan meminum misalnya minuman tertentu. Tadi beliau menceritakan misalnya kopi, kita meminum kopi berturut-turut setiap pagi dengan harapan misalnya akan turun blood pressure-nya. Nah, ini jangan dilakukan ketika kita tidak memiliki data yang cukup terkait hal tersebut. Makanya tadi beliau menyampaikan silakan cari data terlebih dahulu yang memang valid resource-nya, yang menyatakan bahwa kalau misalnya meminum minuman tertentu bisa menurunkan, ya silakan. Tapi kalau kita tidak memiliki cukup data, jangan lakukan itu. Nah, itu mungkin jawaban beliau dari the second questions, dari pertanyaan yang kedua mungkin. Professor, I have interesting questions regarding the first question before. If the diagnosis is accurate and we can do like exception you just mentioned, how long does it take? I mean, the prevention efforts from the patients. How long does it take for, let's say, the effort to reduce their body weight, for the example? Is it really give significant effect? And if it does, how long does it take to the patient?
So I guess the question is how long you should use non-pharmacological measures before you start using drugs.
Yes.
I think this is another important question because this answer is not the same for all patients.
All right. Okay.
If tomorrow I see a patient with diabetics who is 50 years old and he has CKD and he's a smoker, I don't wait a minute. I'll start on the same visit everything I can do to reduce his cardiovascular disease. I'll start with aggressive treatment. I'll start with statins and anything I can do to prevent events. But if I have a low risk patients, and in my clinic, I also see adolescents with high blood pressure. In these cases, you can wait and try to solve the problem with non-pharmacological measures. But there is a trap here. You can't wait forever. You shouldn't wait for 5 years to reduce blood pressure. So I think the guidelines say you can wait up to 6 months in low-risk patients. In my opinion, because I see also adolescents who are overweight, I put a maximum of 1 year because they are very young people, 15 years old, very low risk, but no more than a year in low risk patients. So it depends on the patient.
Alright, jadi tadi saya melanjutkan pertanyaan dari Pak Budi, seberapa lama kita harus menunggu perubahan kondisi, misalnya kita didiagnosis dan diagnosisnya akurat, kemudian kita melakukan prevention tadi, berapa lama yang kita perlukan? Nah, ini jawaban dari beliau adalah bergantung ke kondisi dari pasien itu sendiri. Jika memang sudah bentuknya agak parah mungkin ya, maka agresif treatment mungkin itu yang dilakukan terhadap pasien tersebut. Tapi bila misalnya ada kasus hipertensi pada anak-anak, dalam ini adalah anak kecil misalnya, itu juga kan berpotensi ada hipertensi. Nah, ini beliau menyarankan lebih ke arah non-pharmacological treatment. Jadi itu biasanya beliau menyarankan tunggu sekitar 6 bulan sampai 1 tahun sehingga upaya-upaya non-pharmacological treatment tersebut bisa memperlihatkan hasil begitu. Itu pertanyaannya. All right, thank you, Professor, for the answers. I invite again the audiences if you still have any questions for Professor Sergio, please. All right, probably that's enough from the audience. I still have one question, Professor. Interestingly, this past few months doing some readings, and I have several papers that mention there are several papers mentioning that hypertension still a major problem, especially in low or middle capital countries. This also affect the availability of blood pressure measurement tools in healthcare facilities. So when, let's say, Indonesia is still in developing countries, and do you have any suggestion how can people, especially nowadays with high prevalence of hypertension, to control or to measure blood pressure in such developing countries? Do you have any suggestion? Because some people don't have access to healthcare facilities, even they want to know their blood pressure measurement result or etc. Do you have any, and maybe in your expertise, do you have any practical advice for this issue?
I think this is an important question and the problem we have, and this is why you can imagine how wonderful it would be if we had an application and suddenly all our mobile phones could become accurate blood pressure monitors. We don't have that at the present time. So we have to stick with the methods I discussed, which is office, home, and ambulatory monitoring. So ambulatory monitoring is not going to solve the problem. It's supposed to be the best method. It's not going to be widely available in all countries, and it's not going to be the solution for making decisions in our patients. So we are left with home and ambulatory and office measurements. One thing we should do is we have to standardize our office measurements. There's a lot of variability on how we take office measurements, and we have to make a decision on how to do that. We have to use electronic devices to get rid of the observer, and we have to decide that we press a button and we have 3 readings and we do not talk during the readings. So we have to make these measurements standardized, and as I said, professional devices are expensive, but the properly validated device which can be used in the office is less than 50 euros and can last for 10 years. So the first thing is to optimize and standardize office measurement. The second is for home measurements. I think home measurements is the solution for confirming hypertension out of the patient for most countries. Of course, there is a cost for the patient, and patients have to buy that themselves. The cost is something like 30 or 40 euros, and as I said, this device can last for 10 years. They are very resistant for long-term use. So I think for me, standardization of the office is something we have to solve. And the dissemination of low-cost home devices is the only realistic solution for most patients for out-of-office blood pressure evaluation. And I hope we'll have sometime soon something which is cuffless, but it's not going to come in the next 2 or 3 years.
All right. Yeah. So basically, it's still going to be a big problem probably that we need to address. Yes, Professor. And time flies, and finally we are at the end of the final sessions. It's such an interesting discussions, to be honest, Professor. But as we are at the end of the third session, would you mind give us a closing statement, probably for the audiences, to recap this final
Yes,
session?
I think I am amazed how neglected good quality measurement is.
Yeah.
How physicians often decide to prescribe treatment for years without first having a very careful evaluation of the level of blood pressure. Diagnosis can be difficult in some people's, and more readings are needed. We have no tool to decide who needs drugs for his life or not. We don't have tools to decide who needs 2 or 3 or 4 drugs. We only have blood pressure measurement. So blood pressure measurement is used to make decisions for treatment for decades. We have to take it seriously, and we have to apply the huge amount of knowledge we have and make sure we have accurate evaluation before deciding about treatment. So for me, blood pressure measurement, step 1, before anything, before evaluation, before treatment. You don't need, I have seen patients evaluated for hypertension and treated for hypertension, and when they were carefully assessed, they did not have hypertension. So step 1, blood pressure measurement, hypertension diagnosis, and if you are sure about that, then consider evaluation and treatment. Thank you.
All right, thank you, Professor Sergio, for the closing statement. Bapak Ibu yang kami hormati, penutup dari beliau adalah bahwa yang pertama lakukanlah blood pressure measurement atau ukur tekanan darah Bapak Ibu sebagai langkah awal sebelum menentukan treatment atau evaluasi lanjutannya. Jadi jangan lakukan treatment terlebih dahulu sebelum kita melakukan pengukuran tekanan darah. All right, Professor, thank you for joining us today at the event of our bi-monthly event, actually. And we are hoping that you can join us again in the future sessions, probably, if you don't mind. Yeah.
I hope it was useful for those who attended on this Saturday.
Sure. Absolutely, this is knowledgeable and also useful for us here. And Professor, probably the only limitation for us now is the time difference between Indonesia and Greece. Probably, I believe you are still at 8 in the morning. Yes, if I'm not mistaken. Yeah, we are here at 1 p.m. already. So yeah, it doesn't matter. We still can conduct this meeting together and share your knowledge to us here. Yeah, thank you very much for joining us and giving the very knowledgeable speech to us. And thank you very much. Maybe to close this session, I will ask you to stay in your position because one of our team will take the picture as for the documentation of this event, if you don't mind. All right, 1, 2, 3. All right. Thank you, Professor, for your time again. I hope you stay healthy and stay sharing the good things and doing every good things at your life. We hope we can see you again and meet you again in the near future session. Thank you very much. Hope you enjoy your weekend. I'm sorry for interrupting your weekend, to be honest. This Saturday morning, you already attend this session. Thank you once again, and I hope you enjoy your day, the rest of the day. Thank you very much. I hope you enjoy the day. Yeah.
Thank you, Professor.
Bye-bye. Thank you.
Yeah, I'm handing it back over to our master ceremony. Thank you very much.
Thank you very much, Prof. George Statue and Dr. Ahmad Yani. Ladies and gentlemen, we finally come to the end of today's agenda. Let us close the seminar by reciting Hamdalah.
Alhamdulillah.
We are deeply appreciating all presenters, speakers, moderators, and all participants who joining us in today's event. Those are the agendas we have presented to you all this morning. We hope you learn many useful things today. Please accept our apologies for the mistakes. Thank you so much for your kind attention. See you on the next occasion. Wassalamualaikum warahmatullahi wabarakatuh. Have a good day, everyone.
Warahmatullahi wabarakatuh. Happy
Thank
weekend,
you. Terima
everyone.
kasih, Pak Made.
Yes, Mr. Budi. Thank you again, Professor George Stagio. Thank you, Pak Budi. Thank you, Prof. Kayan dan Bapak Ibu yang sudah hadir. Terima kasih Bapak Ibu dari luar Poltekkes Banten yang sudah berkenan hadir menghadiri webinar internasional kami yang insya Allah akan kami agendakan menjadi agenda rutin tahunan kami bersama dengan 3 event internasional lainnya. Kami tidak sabar menunggu Bapak Ibu di sesi berikutnya di webinar kami di tahun 2023 karena webinar internasional di hari ini merupakan penutup rangkaian webinar di tahun 2022 dan kami berharap dapat bertemu kembali dengan Bapak Ibu di tahun 2023 dengan materi-materi lainnya yang tidak kalah menarik dan tidak kalah informatif dan bermanfaat. Terima kasih sekali lagi, kami tutup. Mohon maaf bila ada kekurangan dalam penyajian yang kami lakukan dan kami sampaikan pada kesempatan hari ini. Bila taufik wal hidayah, wassalamualaikum warahmatullahi wabarakatuh, dan terima kasih. Salam sehat semuanya. Selamat weekend. Informasi untuk Bapak Ibu dan rekan-rekan mahasiswa yang join di kesempatan webinar hari ini, silakan mengisi absensi bagi yang belum karena nanti akan disediakan sertifikat bagi yang mengisi daftar hadir. Terima kasih. Sertifikat akan kami kirimkan ke email masing-masing Bapak Ibu. Terima kasih. Terima kasih kembali Bu Marlina. Sehat selalu Ibu.
Terima kasih kembali Pak, sama-sama Pak.